Thank you so much. Good morning, everyone. Welcome to the International Laurel Wilt Update Research Conference. We have almost 71 participants now, and other people are piling in. So we'll get started. My name is Yuri Hosser, and I'm the forest entomologist at the University of Florida School of Forest Resources and Conservation. That complicated name just means that I run a lab that focuses on forest bugs and forest fungi. But what our team really focuses on is bark beetles, ambrosia beetles, the fungi associated with them, trees they bore into, and people. And some of these people love these beetles, and some hate them. But today, I really want to represent or introduce to you the Bark Beetle Mycobiome International Research Coordination Network. What is that? We are a group of about 30 researchers focused on the symbiosis between bark beetles and fungi. Most of us are from the United States and South Africa. These two countries are where the funding is coming from. But we also have members from Asia and Europe and elsewhere. And our goal, the group's goal, the Bark Beetle Mycobiome group's goal, is to do exactly what we're doing today, which means networking about the research on the relationship between these fascinating beetles and these bizarre fungi and the trees and people they impact. So thank you for being with us. I'm just going to run through a quick introduction here. Some time ago, we realized that we, as the Bark Beetle Mycobiome group, realized that Laurel wilt, obviously, is such a critically important disease. And it really is also one of the sort of flagships of the ambrosia fungus research world. But we, the group, have not really interacted with the community of researchers that study Laurel wilt. There's some overlap, but we don't really interact that much. And that's obviously a problem. So this morning, we wanted to fix that. And we really wanted to connect to people who do the and be people among themselves too, who do the basic fundamental research on Laurel wilt, people who develop applications, and the end users, the huge community of people around the world who need Laurel wilt facts, knowledge to do what they do. Also, I wanted to especially welcome our friends from Mexico. There's a couple of them. Thank you for being with us. You know, we all are imagining probably what might happen if this disease got out of the United States to places like Central America, South America. And we all want to make sure that it doesn't happen. We all want to make sure that we have a forward-looking perspective. And we all want to make sure to do everything we can to prevent Laurel wilt epidemic in the Southern Hemisphere. So welcome, folks. Glad to have you with us. Also wanted to mention, thank you for being willing to do this on Zoom. Isn't that funny that we've been doing this Zoom conferencing way before the pandemic? You know, even this meeting, Zoom conference, was conceived before the coronavirus turned academia into Zoom land. So this has been our new normal for some time. So, and I think it's great because this way we can have many more people attend at the same time. And we all saved at least a ton of carbon dioxide too by not having to travel anywhere. So enjoy the meeting. If you want to keep in touch with us, please join the Bark Beetle Mycobiome group ideally on Facebook. I'm going to just quickly share the screen, which many of you already probably have taken a look at. It's our Facebook group. There is not much traffic. The point is that you get updates when you sign up for the Bark Beetle Mycobiome Facebook group. That's the most important thing. And the one last thing I wanted to mention to everyone is that the presenters will follow the script that's written on the website, our website, the wiki page, and everybody has 3 minutes to present their 3 slides. And after that, there's going to be 3 minutes for interaction. How those work is easy. Almost everybody will be muted except for the moderators and the presenters, but everybody is encouraged to submit questions. through the chat box. So please locate the chat and type in questions for whoever you want to ask a question, especially during their presentation. And the moderators will read those questions out loud, and then the presenter can answer the question. That's the only way we can think of so that we don't turn into a chaos. Okay, well, thanks everyone again. And I will now turn to Romina Gazis, who will introduce us to the Laurel Wilt Working Group.
So hi, everybody. I'm happy to see a lot of familiar faces and some new friends. So Jiri asked me to introduce the Laurel Wilt Working Group 2 minutes ago. So I apologize for being spontaneous. As Jiri mentioned, Laurel wilt is a very important disease to natural forests and also to the avocado production. So it's really threatening not only Florida, but California and other places where avocado is being grown, even though it is only in Florida for now. So University of Florida has a working group that works on extension and on basic fundamental research. This group is comprised by faculty members and extension agents from different departments. So we have forestry people, entomology, microbiology, plant pathology. So we all get together maybe every 3-4 months and then discuss the advances of our projects and our grants that are running right now. So we thought that we are working very collaborative, but only kind of within University of Florida. So this is a great opportunity for us to tell everybody in other universities, as Laurel wilt is increasing and expanding its range, more states are being hit and more universities and working groups are interested in working on this disease and are already working on this disease. So I think this is a great opportunity for us to network, as this is one of the objectives of the network itself. So a lot of speakers from the University of Florida, and we are hopefully put a step forward to expand our collaborative work with others. That's it.
Thank you, Romina. Great. So a bunch of people getting together really for the first time, but some of us are good old friends. Anyway, I will turn the podium to our presenters. Jeff, do we have Jeff Rollins?
Right here, Yuri.
Very good. Welcome. Good morning.
Good morning.
And are you comfortable? And everybody, all the presenters are comfortable sharing their screens, I hope. Looking forward to seeing that. Perfect. It's working. Yay.
How's that? I got
Perfect.
it?
Yeah.
Okay, yeah, thanks, Yuri. Thanks for all the organizers. Great to be able to have this interaction. 3 minutes, so get right to it, right? So my lab's primarily interested from a fundamental perspective of understanding how Raffaella loricola really is such a lethal pathogen here in North America. And we're interested in this both from like an evolutionary perspective and also from a mechanistic perspective. And so I'm gonna tell you a little bit today about our efforts in the area of genomics and transcriptomics to try to get at this understanding. And so this is just a table here just to impress you with the fact that, yeah, we have sequenced genomes. These are draft genomes. We'd like to do better, but as the funding restrictions are what they are, at this point we have draft genomes, but these are pretty good genomes for drafts, and the fact that we feel like we've covered most of the gene space within these genomes. We have about 98% of the conserved predicted genes within our annotations for Raffaella loricola. And so one of the things we're doing is comparative genomics. We're doing it a little more broadly, but primarily we focused on Raffaella aguacate, right? So another ambrosia beetle symbiont that's also associated with avocado and phylogenetically very closely related to Raffaella loricola. And so we have also that draft genome and again, pretty good gene space, about 98% of the conserved predicted genes present. And so we've also have looked at transcriptomic comparisons between in vitro grown Raffaella loricula and inoculated red bait plants. And so within the transcriptomics, probably one of the biggest findings, maybe a big surprise to us, was that we've seen that sulfur metabolism appears to be a really key component of Raffaella loricula infection. And I'd be really interested to know if this is also a component of the symbiosis with the beetle or not. So if you just look at this heat map here, you can see these are sorted by by adjusted p-values as the top 100 most significantly differentially regulated genes. And these that are colored in yellow, the gene names here, about 50% of the top 100 are involved in sulfur assimilation and sulfur uptake, right? And if we look at the broader annotation within the genome, about 150 genes, we see that same trend, that the majority of these are upregulated during infection relative to growth under non-sulfur limiting conditions in culture. So primarily these are involved in the uptake, different permeases and transporters of sulfur and alternative sulfur sources, right? And then their assimilation, their enzymatic breakdown primarily into sulfite, and then feeding into cysteine and sulfur metabolism within the fungus. Right, and so the transcriptomics has also empowered us when we're looking in the genome, because the genome can often be overwhelming with the number of candidate genes that you have, but it's allowed us to look at some of the genes that are differentially regulated from that perspective, also from the whole genome perspective. And one of the things that we've seen looking at all the secreted proteins, if you look at Venn diagram here, agukate and the Japanese oak wilt corsivora, and Raffaella loricula, there are unique genes, secreted proteins within the genome, but again, that's kind of a bit overwhelming. So even through annotation, but combining that with transcriptomics, we really have two real good candidates that we're interested in functional analysis. And the first is a stratoplatinum gene that's unique to loricula and corsivora. And this is known to be an elicitor of plant defense and also a necrotrophic effector, depending on the context. And then the second gene is a gene encoding a protein for an aerolysin-like protein. And these are well-known pore-forming toxins found in bacteria, found in some fungi, found in animals as well. And basically they form pores within membranes and allow for leakage. Okay, so these are basically putative effectors, those things that are acting as virulence determinants for this fungus. We've also focused on secondary metabolites with an eye at looking for toxins. And the one thing you can see here from this table, comparing loricula to agukate, is that there's a massive expansion of secondary metabolism capacity or potential within loricula. There's 37 secondary metabolite gene clusters relative to 27. And if you look at these, they're unique both in agukate and loricula, but you find this real large expansion of both the polyketide synthases and non-ribosomal peptide synthases, and their hybrids within loricula. And this is just one example, one that's upregulated in planta. We call it cluster 7. It's a polyketide synthase. It's also found in Pestulodiopsis phyci. It's very well conserved within Pestulodiopsis phyci, but not within agukate. In fact, the entire gene cluster and upstream and downstream genes of that cluster are absent from agukate. And then a second cluster, which is a PKS NRPS, the same thing, the functional part of this cluster is missing from agukate. So these are two, again, candidate clusters that we're interested to see what role they may be playing in Raffaella virulence. Got about 3 minutes.
Thank you, Jerry. So should I read the questions that being asked from the chat box?
Absolutely. So just to explain, Tuan is one of our moderators, and Tuan, it's up to you. I'm sure you have your own questions, or might be worth just sampling the questions that have been piling up in the chat box.
Yeah, because we have 3 minutes. So let's get the opportunity for other people asking questions in chat box first. So hi, Jeff. This is Tuan here from Fabi in South Africa. So thank you for the talk, and hello for everybody. So I'm just going to go to some of the questions. If there isn't time to go to all the questions, I apologize. You can go to it later and maybe ask Jeff personally yourself. So Jeff, there's a question from Jerry that when you say that agukate is associated with avocado, does it mean that it's native to America? So
I'm not sure. You've probably done more. You have done surveying. I know that it's been isolated. It was the first potential Rafflesia that was isolated from avocado in Florida. And it's been subsequently isolated from avocado. And if I'm not mistaken, and it's been isolated from ambrosia beetles. I don't know the species. There's others here that have done this work that are involved in that, and they could probably expand on that. If I'm not mistaken, it's also one that's been found in Mexico, but I'm not sure of that.
Okay, and yeah, thank you. And then there's the follow-up question that what beta host the aguacate isolate from? So what vector is it? What vector is the fungus?
So again, I think there's others here that have actually done that work that could speak of that.
Okay, I have a question myself on the RNA-seq data analysis. So you said that sulfur metabolism is upregulated in Loricola. Do we also do that do you also did it on aguacate to see, is it the same thing, or is it like more Rafflesia thing, or is it specific to Loricola?
Yeah, so those are things that we'd really like to dig into, but haven't been able to do at this point. So we'd like to do an expanded actually RNA-seq of infection of avocado, and to take more time points and see if this trend continues, or if other things, actually metabolites, metabolism, and putative effectors come out and are more important, or at least the dynamics are shifting during different points of infection, but it's not something we've been able to do at this point.
Thank you. And then also the point on the expansion of secondary metabolite gene cluster. It could be expansion in Loricola, but it also could be construction in aguacate. So maybe look a little bit broader on the evolutionary history of the whole related fungus surrounding it. And Jiri also have a question that isn't Quisivora is from a different clade to,
absolutely.
Yeah, it is actually a different clade, that's correct.
Absolutely. Yeah, we were kind of interested in some of the broader and some of the other Ophistomatales as well. Really wasn't presenting a lot of that, but some things that may be conserved that are pointing towards a conserved evolution towards pathogenesis, and then those that are probably more lineage specific that may result in, like host specificity and what have you. So that's taking both a close look and a more broad look. I think both are important within that broader lineage.
Right.
Regarding aguacate, it was from a dying avocado originally. We isolated it originally from a dying avocado in South Florida, but confirmed to not be a pathogen multiple times. And it is extremely close to Loricola, of course, but never known from any beetle that I know of from anywhere.
Yeah. Thank you, Jason. And there's a question from Danieta. What are the time point that you used for the experiment, I presume, for the RNA-seq analysis?
Yeah, so that's basically a single time point shortly after inoculation. So at about 3 days post-inoculation, and it covers the inoculation point and above, and about 5 centimeters above and below the inoculation point.
Thank you. Let me just quickly go through the chat box to see if another question. No, I haven't seen any.
There's one excellent question from Louise.
Yeah, that's right. Yeah, so go ahead, Jerry. Go ahead.
Knowing that these sulfur enzymes are in the infection process involved in it, would they make good targets for a more applied work? Is it conserved to Rafflesia itself, and what's the relationship between what you know about the sulfur enzymes and application?
Yeah, so Chung will speak a little bit about our initial work that we're doing to characterize the sulfur assimilation, uptake and assimilation pathway in Rafflesia, but it's highly conserved within fungi, right? And it's basically an alternative. It's a regulation of the utilization of alternative sulfur sources. So when you're under sulfur starvation conditions, you turn on lots of these uptake and assimilation transporters and enzymes that allow you to basically scavenge sulfur. So we've thought a bit about, the practical application of that, and you could envision both. It's conserved within fungi, but it's not something, that pathway that's known outside of fungi. So that actually makes for a pretty decent target from that perspective, because a lot of development of pharmaceuticals or fungicides aren't towards specific organism targets, but rather broad targets. broad numbers of fungi. So that could be one approach. And the other is just how trying to understand this dynamic of the sulfur utilization, right? It's primarily sulfate that's taken up in the xylem, and that would be the primary sulfur source, probably for Rafflesia, but it appears to be able to scavenge other sulfur that may be within the walls or in the parenchyma associated with the xylem. So we're trying to get an understanding of that dynamic really, and then seeing how maybe exogenous sulfur and uptake of sulfur or excess sulfur, perhaps if it's a battle between the fungus and the tree for available sulfur, then perhaps there's ability to actually provide the host with enough sulfur that battle ends, and maybe there's more ability to defend, et cetera. I don't know. It's all pie in the sky at this point, but we're trying to get a better understanding of what that sulfur dynamic and the essential, whether it's essential or not for pathogenesis.
Thanks a bunch. Excellent. We're gonna jump to Josh. Before we do that, I just was reminded of two things. One is that this whole thing's being recorded, and you will all get the link, and you will all be encouraged to send it to whoever you think might be interested. This is all very open, serving the world. That's one thing. Second, we'll do a quick break between the sections ecology and detection, just so that we can, catch our breath. Josh.
Hey, how's it going? Hear me okay?
Good morning, yes.
Hey, good morning. Oh, great. All right, go ahead and share. Okay, I'm here to talk to you guys about a colonization of swamp bay by Rafflesia auricola, which is a project that I'm working on right now. It's actually still in progress. I'm still making observations. So to start out, Chung Wang, who's going to speak after I do, he created this great fluorescent GFP strain of Rafflesia auricola, which is very, very, very bright. And most importantly, when you inoculate a plant with this GFP, it doesn't turn off the GFP, which is a problem we had before, is that the GFP would turn off in planta. So this one did not do that. So we inoculated swamp bay with 3,000 propagules or CFUs of the pathogen with the GFP. And we did this with spatial and temporal sampling. So we did these different time points, these different days after the inoculation. And then we would take those different sample points at those different times, and we would sample 10 centimeters below the inoculation point. And then we would take samples in 10 centimeter increments along the stem from above the inoculation point all the way until the end of the plant. And we also did the same thing for branches. We wanted to look at the dynamics of what things look like within the branches as well compared to the main stem. And these are all with 3-gallon trees in the greenhouse. All right, so then we took these 10 centimeter segments and we stored them in 4% paraphenaldehyde. And we were able to, we sliced them using a microtome into 25 micrometer sections. So for each section, we would make four sections for each 10 centimeter segment. And then these were made into slides and they were observed in their entirety under the fluorescent microscope. And what that enabled us to do then is to sort of get an overview of the quantification of the different CFUs within the tree. So we could look at different time points and different distances along the plant stem and sort of get an idea of and quantify what we were able to find there, as well as look at sort of the morphology and how the pathogen was colonizing within the plant. And then the next stage will then be sort of comparing the quantification at these different time points and different intervals along the plant stem with histological observations so that we can gauge the host response from the plant as well to these different levels of the pathogen and where the pathogen is throughout the plant at different points in time. And so one of the other things we can do then with the quantification data is we can start to sort of get a general idea of the estimate of the colony forming units that are in a tree. So it's not going to be, a perfectly accurate measurement, but it's good for making comparisons at the different time intervals as to how the pathogen is building up within the tree over time. And so you can see this is the experiment's still in progress at the moment, but we have up to 21 days at this moment. And you can see that the increasing levels of the pathogen at different time points. And then I also put on there the mean disease severity rating, which is a scale from 1 to 10, 1 being healthy and 10 being totally dead. And then the other thing we can then begin to do, and this is sort of a preliminary placeholder graph that's going to be improved later on, is kind of get a general gauge of where the pathogen is in the plant at different time periods within the infection process. So we can sort of get an idea of how far along, vertically along the stem, you're going to find the largest amounts of pathogen. And what we found mostly is just that a large concentration really begins to build at the very end of the plant. The pathogen very quickly moves to the upper portions of the plant. And I think the measurement we have right now is it's 22 centimeters 22 centimeters per day is the average movement rate. But these were in small plants, and really that was limited more by the height of the plant than anything else. So I think that within larger trees, it's actually moving much faster. So this pathogen can really move very quickly throughout the xylem of the tree. And that's all. I'll move on to questions.
Okay, thanks for that great talk. I'll give people a few seconds to write their questions. So one thing that Jason asked is if you looked in the leaves.
Yeah, so we did play out some leaves, and we have some leaf sections. We need to get the cryotome working so we can sort of do some sections along the leaves and see what we can find there. But we were able to recover from the leaves beginning from 10 days after inoculation.
So Andrew, can I ask a question? Sorry.
No, go on. Yeah.
Very nice, Josh. I'm quite curious as to in what form the fungus is actually moving through the tree. If you're saying CFUs, is it just mycelia, or is it actually sporulating inside, or is it changing into a different phase?
Yeah, so that's one of the things I'm looking into more detail right now. There's a lot of interesting morphological features that we sort of still need to identify. But at the moment, it looks like a lot of, it seems to be moving mostly by these, a lot of blastospore movement, sort of like yeast-like cells, as well as spores early on. And then those begin to germinate into hyphae. But the hyphae really isn't penetrating so much into the parenchyma so much as it's actually crossing over into adjacent vessels and colonizing those. So there's a lot of spore movement and not a lot of mycelia early on. And then as you start to see mycelia, a lot of that's just penetrating into adjacent vessels. And so you can actually, this photo at the top right here, you can see that where it's going from one vessel over into an adjacent vessel, which seems to be the first phase.
Okay, there's been a couple of questions relating to using swamp bay and how this could relate to avocado, or if you have any plans to carry this out in avocado plants also.
Yeah, we do plan on, that's going to be the next step is moving on to avocado next. So one of the reasons why we started with swamp bay is, quite honestly, they're just cheap. They're much cheaper than avocado plants. So, and Jason Smith was really nice and allowed us to use a lot of his swamp bay trees that were clonal susceptible trees. And so, yeah, so there's a lot of them, and we were able to buy a bunch as well from nursery for much cheaper than what we could for avocado plants. So this is sort of like our test model to start, and then we'll probably progress onto avocado afterwards.
Okay, so lastly, I guess one of the big problems with avocado is transfer through roots. So did you also go into the roots and do the same downwards?
So we didn't go all the way down into the roots, but we did sample 10 centimeters below the inoculation point to sort of get a general gauge of how it's moving below the inoculation point and about how early on that begins to occur and what form we can find it. So so we did start sampling 10 centimeters below, and we were able to recover 10 centimeters below, but not all the way down into the root system. I believe Ramina's working on that. She might talk about that in a little bit.
Thanks, everyone. There are many more questions. It's amazing, but we sadly don't have time to answer them all. We're gonna move on. One last thing for the presenters, if you can just in one word say which institution you're from, that would be nice. Dr. Chung Wang, please.
Okay, good morning, everyone. I'm very glad to share my current work. I'm going to talk about using CRISPR-Cas9 technology to knock out of genes in Rafflesia lauricola. As it's well known, gene knockout is usually difficult on non-model organisms like Rafflesia lauricola. However, with the development of the genome editing tools, CRISPR-Cas9 is a very promising tools for gene function characterizations. I will introduce this system using two examples. Previously, we found a very good promoter to drive the GIP. We can easily find the GIP fluorescence in the protoplast during the transformation. So we want to try using this promoter to drive Cas9 gene transient expression and a 5S guide RNA to drive guide RNA expression, transcription. So to check our system works very well, we first choose the URE3 gene to knock out, which is quite a functional, conserved, and a very well-known negative marker in fungal genetics. URE3 can transfer a non-toxic substrate 5-FOE to a very toxic substrate 5-fluorouracil, which is quite toxic to RNA biosynthesis. So URE3 mutant can grow on 5-FOE medium. So this is our transformation plate. When we add more, during the transformation, when we add more Cas9 at guide RNA plasmid, we found more colonies show on 5-FOE medium. We randomly choose the 3 mutants for sequencing. We found all of the target sequence had short deletions, a nucleotide short deletions, causing the URE3 frame shift, disrupting the URE3 function, which is a typical feature of the CRISPR-Cas9 mediated gene mutants. And the left panel shows the fungal growth. We found the wild type and the URE3 mutants grow on the different medium. The wild type can grow on the minimum medium, but when we supplemented the uracil and the uridine, all the strings can grow. But when we add the 5-FOE chemical, and only the mutant string can grow. So this experiment means our system works very well. Our RNA-seq data previously showed that Rafflesia lauricola during the infection can massively upregulate sulfur-related genes. So here I want to investigate how the fungi use the sulfate compound. So a knockout of a transcription factor responsible for the sulfate assimilation pathway. We call it this gene is the MET-R gene. I choose and this the MET-R genes on the chromosomes. It contains the 2 exons with a large entrance. I chose a targeted sequence at the 5' and expected using the hygromycin to integrate this site and disrupted the normal function of these genes. I added the 14 nucleotides homologous arms on both ends of the hygromycin. After transformation, we selected 2 mutants, and this the fungal growth on the minimum medium. We can say the mutant cannot grow on the minimum medium, but when we added some sulfur-related amino acids, the mutant restored growth. That means the mutant cannot use the sulfate to biosynthesize the amino acids. And we also did the pathogenicity assay using the swampy trees. We inoculated 3,000 conidia of these different strains, wild type, mutant, complement strain, and this is the symptom after 4 weeks. And we found the wild type and complement strains can cause the typical Laurel Wilt symptoms like the whole tree becoming wilt, a brown color xylem. And that means these genes lead to is involved in the pathogenicity assay. So yeah, thanks to my PI Jeff Rollins, thanks to my lab members, thanks for your attention.
Thank you very much, Wang. So let's wait for the question to come in. But while waiting, I have a question. So what is the transformation efficiency of the system? Did you calculate that?
I use the PEG-mediated transformation. So in detail, so we usually use the germinated cornelia and using the enzyme to remove the cell wall to form the protoplast, and then we can transfer the plasmid directly using the PEG. Yeah, we can use this promoter to drive the GIP, and during the transformation, we can see the fluorescence. That means the transient expression. It is very good.
Okay, thank you. And then there's a question. Dan is asking that can he see the first slide again? I'm not sure if you can share the screen again for the first slide, please.
Sorry, say it again.
Dan is asking, can you please share the first slides again? The first slide of your talk.
First
slide.
First slide.
The first slide of the presentation. Would you be able to share it again, please?
Okay. You mean this one, is that right?
That's right. That's right, Dan. You go ahead. Do you have a question, Dan?
Yeah. So I use the promoter. That promoter is called elongation factor 1 alpha promoter. It is very, very strong promoter to drive the GIP. You can see this picture under the title. And I use this promoter to drive the Cas9 and the 5S promoter to drive the guide RNA. We can do the transient expression. Because when we knock out the genes, we only use these two components. That is the guide RNA cassette and the Cas9 cassette. And during the transformation, we can see the mutants. The mutants, it showed there's nucleotide deletions at the target sequence. That is a typical feature of the CRISPR-Cas9 mediated gene deletions.
Okay, thanks, John. Let's see. Okay, so can I ask a question then? So which promoter did you use to drive the hygromycin gene?
Hygromycin. Hygromycin, we used multiple versions. That is the TRPC, GPDA, the promoter. All of these two promoters I tried works very well.
Okay, and then I have a question. What is the MM supplemented, so mineral supplemented that you get to grow on? So what is the supplemented? With that, you get to grow on.
The methionine and cysteine, you mean?
For the MM, I guess that was white tie, and the last one is a complementation.
Oh,
okay.
MM, that's the minimum medium.
Yeah. Okay.
Using the massive element and the minimum elements. Yeah. Right. Yeah. I can send you the recipe of this minimum medium.
All right, good. And there's a question from Joseph. Is the expression of Cas9 transient because the promoter is inducible? We
not changed much. This is a version from previously publication. So we request the plasmid and clone it from that plasmid. Yeah.
Joseph, I think it is transient because I mean they use the same one to drive the expression of GAP, and it's supposed to it's not supposed to be inducible promoter if I got it right. Okay, Jerry. Thank you, Jean.
Thank you.
Thank you. Fantastic. Let's move on. Ross, you're on.
Okay, great. Just sharing my screen now. Can you guys see that okay?
And please, again, if the speakers can tell us which institution they're with.
Okay. Just give me 1 second. Okay. So hi, everyone. My name is Ross Joseph. I'm a second-year PhD student in Dr. Nemat Kehani's lab at the University of Florida. And today, I'll be telling you a little about some of the work that our lab is doing to facilitate molecular and genetic research into Raffaellia lauricola, and hopefully be able to probe its ability to infect both plant hosts and to form symbioses with its insect vectors. So for scientists to begin to dissect and explore the molecular mechanisms that mediate biological phenomena like host microbe interactions, we first need to be able to transform our study organisms efficiently, and we need to be able to discern between the transformed organisms and wild type strains. So in Chung's talk, which was great, he was using fluorescence and also a toxic medium. And so that's how people kind of commonly do this, is they introduce what's called selective markers. And we wanted to kind of see what the field of selective markers that we could have to use from were. And so based on this idea, we performed a resistance marker profiling in order to identify drugs that Raffaellia lauricola is sensitive to. And this is simply done by growing the fungus on plates containing different drugs at different concentrations, and you can see how these affect the growth of the fungus. So here we have shown 5 drugs that we screened. And using this approach, we found that drugs like phosphenothricin, hygromycin, chloromirin ethyl, and benomyl will be effective selective agents because they're able to fully inhibit the growth of wild type fungus, whereas the fungicide G418 was not able to fully inhibit the growth, and so it wouldn't make an effective selective marker. And so we could then take this information and go forward, and we designed a number of plasmid vectors using the BAR and SIR markers. So these code for resistance to phosphenothricin and chloromirin ethyl respectively. And we also used both exogenous promoters and endogenous promoter elements from Raffaellia lauricola to drive the expression of these resistance genes. And these allow for the efficient transformation and selection of Raffaellia lauricola mutants using both agrobacterium-mediated transformation methods, which you see in these 2 plasmid maps on the bottom here, and also a lithium acetate-based method, which you see in the 2 plasmids on the top here. And these transformation methods yielded 1 to 200 colonies per 10 to the 5 spores, and 5 to 60 colonies per microgram of DNA per 10 to the 8 spores, respectively. And so on the bottom here, you can then just see kind of demonstrations of how these transformation plates look. These mutations were found to be mitotically stable after 5 generations, and greater than 95% of the mutants that we screened were found to be the result of a single integration event, which is nice. So then using these agrobacterium vectors, we created some different reporter strains that stably express GFP, RFP, and GUS over here. So GUS just turns blue when you add a certain chemical to it. And these agrobacterium vectors, we could also use them as random integration vectors, and we used those to screen or to generate a large library of random insertion mutants in Raffaellia lauricola, which we could then use to screen for different phenotypes to see mutants that are involved in different phenotypic processes. So these reporter strains and the mutant libraries that we generated represent powerful tools for probing the molecular mechanisms of both virulence and symbiosis that this fungus is able to exert on its plant and insect hosts. And so currently and on into the future, we're hoping to use these tools in future studies to elucidate the mechanisms of symbiosis and virulence that Raffaellia lauricola uses with its host organisms. So thank you. With that, I'll take questions.
All right, I'll allow anyone trying any new questions, but I was wondering if earlier in a talk, someone mentioned how some of the fluorescent strains seem to be like unstable, or at least like no longer fluorescent in different parts of the plant. For your various new strains, have you tested that? And how do you go about finding that out?
We've done some preliminary work with looking at colonization in the trees using the reporter strains that we have. That work was mostly done by a former postdoc in our lab, Yang-Hong Zhu. And we did find some difficulties in visualizing based on fluorescent signal in the plant. We are currently performing studies where we look at colonization in the mycangia of the ambrosia beetles by these fluorescent reporters, and we're having some more luck in that system using these methods. But it's still pretty preliminary work, so we decided to hold it out for the purposes of this talk.
Okay, I just had a question from Chase Myers. What kind of symbiosis mechanisms with the beetles might be tested with these new strains?
One of the things, well, first off, the mutant libraries that we generate, so they're all conceivably single deletion mutants where kind of a gene in the fungus's genome has been knocked out and basically replaced with one of these fluorescent reporters. And so we basically have a library of mutant fungi that are also fluorescent and knocked out in a single gene. And so we can use these to screen against the fungus, or screen against the beetles and look at their ability to colonize the mycangia following feeding. So we can ask the question, are there mutants that are impaired in the ability to colonize? And then we can go back and look and say what genes are responsible for these observed phenotypes.
Okay, we have a couple of questions from Yuri. Firstly, can this be used to make antibiotic-resistant strains so we can rear these beetles without problems of contaminating fungi?
Yes, conceivably. I've thought about this before, and I'm not exactly sure what the effect that these drugs that we tested would have on the beetles if you put them in the media and grew it. But I think that, yeah, you could generate like antibiotic resistant strains of the fungus, and then you could supplement the media with the antibiotic and conceivably, you know, inhibit or dampen the growth of other wild type organisms that you get in there.
And so Jason Smith has asked, is there an advantage for visualizing the signal in the beetles versus the host?
Does he mean the tree host?
Sorry, the host.
With regard to the different reporters. So in other words, if you use red fluorescent protein versus the green fluorescent, you know, would one, you know, work better in the beetle versus the host plant? You know, I'm thinking like, you know, we already have a GFP strain that seems to work quite well in the host, but, you know, we're not sure about in the beetles. And I'm thinking perhaps you might get a better signal that would work better for visualization of the interaction in one versus the other.
Yeah, definitely. So just in some of the work that I've done, and I'm sure other people who have worked with beetles and tried to do microscopy in the past can kind of corroborate that the exoskeleton of the beetle and a lot of the tissues in the insect autofluoresce, and they tend to autofluoresce at certain wavelengths. And so using that, you might be able to tailor your study to say maybe, okay, we get a lot of native autofluorescence in the, like, say for instance, the GFP wavelength. And so let's use one of these other reporters in this study, different things like that.
Right, thank you.
Okay, we have one final question from Luis. And so using the GFP model, could this be used to visualize the fungal, but not the infection, but like the growth in the beetle?
Yes, that's work that we're currently doing in our lab, and we're it's still a little preliminary, and so I decided to not show too much results from that yet, but that's work that we're actively doing right now.
I'm hoping to see this at the next installment of the Laurel Wilt.
Yeah, absolutely. I'm hoping to get it out there as soon as I can.
Thank you, folks. This concludes the molecular biology section, and let's take a breath of fresh air and move on to basic biology. And we're going to start with Daniel and his super productive, crazy productive research lately. Do we have Daniel Carrillo?
Good morning. Give me 1 second. Okay. Good morning, everybody. My name is Daniel Carrillo. I'm with UF in the Tropical Research and Education Center in Homestead, in the middle of the avocado producing area, and also in the middle of the Laurel Wilt epidemic in avocado. So I'm gonna try to give you some context and give you 8-year story in 3 minutes. So initially in avocado, there was a great deal of preparation for Xyloborus glabratus, the primary vector of this disease. A lot of work preparing for its arrival to the avocado producing area. But it happened to be that the Xyloborus glabratus never really colonized the avocado groves. This beetle is extremely rare in avocado systems. And perhaps this is one of the reasons why the epidemic in avocado has been really damaging for the avocado growers, but we don't see this devastation that is observed in the natural forests. So we have a situation in which this primary vector is very rare, but we found out that several other of the local ambrosia beetles can carry the pathogen. And some of these species can transmit the pathogen to avocado. So in the past few years, we have made multiple studies to identify the symbionts of these species, study their basic biology, their behavior, and also come up with management strategies for the avocado growers. So I wanted to point out this very recent study that is gonna come out really soon. We were studying not only at the mycangial symbionts, but also how they carry this pathogen phoretically on the exoskeleton. And we have found that this is also a potential mechanism for movement of this pathogen. Several of these beetles can carry the pathogen also phoretically. And we are right now focusing a lot of our attention on this species, Xyloborus bispinatus. That is the species that is most frequently found associated with the pathogen in avocado systems, that also in the transmission studies seems to be one of the most efficient species transmitting the pathogen to avocados. It's also a species that seems to have a great flexibility in its symbiotic associations. There are published studies showing that the beetle uses different symbionts in the different life stages, and we still don't have a clear idea of the symbiotic associations of this beetle. So we are studying in detail, and Octavio is going to focus his research, his PhD. He's going to be talking next about this beetle. So I guess that's what I can tell you in 3 minutes. I'll be happy to answer any questions.
Thank you, Daniel, for the presentation. While we wait for some questions to arrive, I actually have one. So you mentioned that glabratus is not common in avocado, but even if it's not actually breeding there, have you seen some signs of infestation or attack, or even when the tree is pushing out the beetles, maybe that might be a reason why the initial tree starts to die with the introduction of the pathogen?
Yeah, initially we studied that in detail. We were working on two hypotheses. One, that we have recurrent introductions of glabratus from natural areas that are surrounding the avocado growing area here. And we conducted some spatial analysis to see if that hypothesis would correspond to the spatial distribution of the disease. And we have a very focal distribution of the disease, showing that is mostly an internal factor that is playing a major role in transmission of the disease. We haven't seen the beetle attacking or breeding in avocado trees here in the growing area. I have seen it in Myanmar, in other areas. I've seen glabratus attacking avocados, but not here. We've looked at the extensive sampling, not only my lab, multiple labs have been looking, and it's extremely difficult to see avocado glabratus associated with avocado.
Thank you, Daniel. I have a question from Yuri. Ever seen bulbulus or bispinatus colonizing living trees?
Yes, this is also an important area of research. So, you know, most of these beetles are regarded as secondary colonizers, or colonizing only dead or dying trees. But what we have seen here is that this is not a black and white situation. We observe that sometimes the trees are subject to a particular stress factor that is not related with Laurel Wilt, but that other stress factor induces the attack by these beetles. So usually, most of the trees that you look that are apparently healthy, they have interactions with ambrosia beetles. It doesn't need to be a dying tree.
Thank you. Daniel, I wanted to give you an award of the speaker that was the first speaker that was on time. Thank you. I would like to encourage all speakers to stick with the 3-minute limit. We're already a little bit behind, so let's make sure everybody has time to talk. Thanks a bunch. Octavio, are you with us?
Yes, I'm here, sir. I think I cannot, there it is. Okay, let me share my screen. Where it is. Oops. Let's see. Can you see my screen there?
Yes.
Okay. So hi everyone, my name is Octavio Menacol from the Tropical Research and Education Center at the University of Florida, and I'm gonna be talking about a little bit of my project, my PhD project. So as Daniel mentioned, Xyloborus bispinatus is consistently found in avocado orchards here, and it has been found carrying the Laurel Wilt pathogen. It's interesting how, based on the amount of information and what we know so far about Laurel Wilt, there's little information regarding the interaction between this beetle and the pathogen. As you can see, like a couple of papers that I have put there, it's basically they have studied the interaction between bispinatus and Lauricola. For my PhD, in particular for this chapter, I would like to address a couple of questions, specifically determine the effect of Rhaphiella lauricola and other symbionts on X. bispinatus fitness, and determine if prior exposure to a given symbiont predisposed X. bispinatus feeding habits. So basically for this, I have come up with two big experiments. The first one, it's basically the establishment of fungal monosymbiotic colonies in which we want to see the effect of each symbiont on the beetle's progeny. So in this case, and it's a research that I have ongoing right now. So basically we collect the pupae, we try to sterilize them with different ingredients, different materials like bleach, ethanol, sterile water. We expose those pupae, or we rear the pupae in different symbionts. Then after complete sclerotization and hopefully copulation, we then move them to an avocado sawdust media. Then after 40 days, we dissect these colonies, and then we record the different response variables, in this case, like eggs, larvae, pupae, and adult. Obviously, we have a subset of beetles that we process, and we play the aliquots in PDA and CSMA to see if there is like no contamination by other fungi. So basically, I'm gonna be like discarding those that present more than one symbiont. For the second experiment, this is basically a factorial treatment design, and this is basically to test whether previous exposure of the beetle of a beetle to a symbiont affect their feeding habits. So I have come up with this binary choice test in which we're gonna have like different colonies of Bispinatus with a particular symbiont, and then it's gonna be exposed to, or we're gonna give them the choice to pick either the same symbiont in which it was like reared on, or a different symbiont. This is experiments that it's ongoing right now, so it really didn't make any sense to share those results yet, but you're gonna see them eventually. That will be all. Thanks.
Octavio, thank you so much again. Thanks for being on time. This is great. Wilma had an interesting question. I'll try to boil it down. So do you have Loricola involved here? Loricola being the accidental symbiont from Asia. Do you see it dominating naturally the Mycengia or the colonies of these American beetles, or is it always just tacked on as a sort of a carry on?
Okay, so not really. Basically, so we have done some surveys, and we have found that it's like in the case of Bispinatus is more 50% of the chances we have like 50% chances to find Loricola either phoretically or in the Mycengia compared to other beetles. Like for instance, you can see Loricola in Volvulus or Affinis, but in like really low percentage compared to Bispinatus. Obviously, the three species are really common in avocado orchards.
Interesting. Sort of a related question from Chase Myers. So do you, in your experiments, do you include subsequent transmission tests? In other words, do the beetles, does the fungus persist on the next generation of beetles, and does it get carried over to the next generation of your colonies?
Okay, yeah, I didn't want to put that one, but here's the thing. So every time that I do like the cleaning process and I feed them, I rear them in an sterile artificial media. But then I'm evaluating the effect of that particular symbiont when I'm sure that it's the only fungal symbiont in the Mycengia. I've been reared, or I want to rear them like over time, basically, like to see the effect of those symbionts through generations. I have like right now, that's actually a really interesting thing. I have not, I don't have in mind like a subsequent transmission test for this one, but certainly that will be like very useful. Yeah.
Thank you. And the last question from you, Lee. Your lab has isolated some interesting yeasts from these beetles. Do the yeasts that you end up isolating in large CFU counts, do they feed the beetle? Do they sustain the beetle alive without the Raffalias?
That's an interesting question. So basically, my experiment, it's only limited to well, fungal symbionts. In this case, like from the Raffaella genera. So I haven't like really tested other yeast. they might probably have some sort of function in the beetle's reproduction, in the beetle's feeding habit. But I have not tested like any specific yeast in my experiment.
Excellent. Thank you, Octavio. Thanks for being on time. Let's move on to Kirsten. Hello, Kirsten. Good morning.
How are you guys? Let's see if I can share my screen here. Oh, somebody's still sharing. There we go. Let's see. Okay. There we go. Can you see it?
Mm-hmm.
Hello? Oh,
yes, absolutely.
Oh, great. Okay. So, you know, this is kind of a small project that we've been working on for a few years that I'm going to talk about. We've been looking in my lab and also in collaboration with Dan Carrillo's lab at characterizing the effect of the microbial community structure. So here, mostly the fungal community, but recently the bacterial community. Looking at how the composition of that affects the transmission of Raffaella loricola. So our objective is really has been to kind of determine whether or not this affects, you know, how loricola establishes in the mycangia of the beetle. And so what we've been doing is to look at, we've been using Xyloborus bispinatus and volvulus. Basically what we've been doing is disinfecting all of the pupae and then rearing them on sterile diets that have been inoculated with single colonies that have been isolated from the beetles. So there are a number of different Raffaella species that we've been looking at, and then seeing how the previous exposure to these different isolates affects subsequent acquisition of the loricola. So we leave these beetles in tubes of diet that's sterile with these different isolates for about 5 to 7 day acquisition access periods. And then we do the transfer for another 5 to 7 days. And then we transmit or give them some time to transmit to a diet only treatment. So they can dig the galleries and then eventually we collect the adult beetles and do the extractions for fungal community analysis. And we've also done some plating as well in these experiments. And this is just kind of laying that out a different way. We've been doing some sequencing of the ITS gene sequences that are associated with the different fungal groups. And I have these slides out of order, but we do see a pretty big abundance before we do these assays if we're testing the beetles of multiple Raffaella species. And we're still kind of analyzing the data, but we do see a certain percentage of the beetles are infected with the different species that we're inoculating them with prior to the Raffaella, but loricola. So it's a little bit difficult to get completely clean colonies of the beetles we found. So what we've also been trying to focus on is to see whether we can look at the associated bacterial communities that are in the mycangia and see whether or not these are also affecting acquisition and replication of loricola in the mycangia. And so for this, we're simply treating the beetles with antibiotics, and we have a kind of a blend of antibiotics, tetracycline and rifampicin, that we're using to clear the mycangia. And then we expose the beetles to Raffaella loricola, and then we're using 16S and Illumina sequencing to kind of look at these different results. So those are two kind of tracks that we're using to assess how these microbial community structures can affect that transmission. And so this work is still ongoing. And I'm going to leave it there for questions because I'm right at 3 minutes. That work.
Right, thank you, Kirsten. So we have one question so far from Ross. How do you go about disinfecting pupa prior to transferring to rearing tubes?
Yeah, so that's been an interesting process, and Alicia Kelly is on the call also. So she's the student that's been working on this. So we go through a series of surface sterilizations of the beetles. So bleach, several washes with distilled water in between. We've tried some other chemicals which will work, but we if we move sort of beyond the bleach, we end up with a lot of mortality. We get quite a bit of mortality anyway when we're sterilizing the beetles. So the bleach sterilization with multiple distilled water rinses is what we've been using.
All right. Any other questions? You can also raise your hand if you have one. Do you do these experiments at different temperatures, considering that the fungi might prefer a different temperature or humidity?
Oh gosh, that's a really great question. You know, that would be really interesting to do, and you're absolutely right. But just the difficulty in getting sufficient insects to do this just at one temperature is about all we can handle right now. I mean, you end up with a lot of things to analyze, and it's quite expensive also to do the analysis. So I think once we have this, you know, done kind of at the biologically relevant temperature, then we can start to play around that mean.
And then we have another question. Are you pooling your samples?
Yes, so that has been something that has come up recently, especially for the 16S work. We simply have to just to get a sufficient sample. So we tend to pool all of the samples in a single tube. So we use multiple beetles per replicate and pool all of those together. So we have to run quite a few trials of this to get sufficient DNA to do the analysis.
All right, so I think we'll take one last question, and that's from Chase. And he says, how do you avoid the ITS amplification problems? Or did you use the ITS2 primers?
I believe we are using the ITS2 primers. Alicia, can you nod or give a thumbs up to that?
Do you want me to answer?
Can you talk? Yes, that'd be great.
Sure. So I don't know that we did avoid it, but I found a publication that has ITS genes that's kind of, I don't know, better. They were, I want to say ITS7 and ITS4. So they worked a little better, and using like a high fidelity polymerase helped. But there probably still was some amplification bias towards the species that amplify ITS a lot better.
All right, thank you. Thanks for that. I think we are at the end of our biology section.
That is correct. Thank you very much, everybody. Does it look like we should take a 5-minute break now, or should we jump to the ecology section? What do you feel like? No break? Let's keep plowing through. Sounds great. Anita, hello.
Hi, guys. How are you?
Welcome.
Thank you. Thank you.
And can you tell us who you are, where you're from? Thank you.
Oh. University of Tennessee. Can you see my slides?
Absolutely.
Okay. So we are new kids on the block in Laurel Wilt research. So this appeared basically last year in Tennessee. And the projections were that Laurel Wilt will move roughly in 2020 to 2030 in this part of the United States. That did not happen. Let me move my slides. So for those of you that are not familiar with our state, it's circled here in purple. And these are the counties that were basically confirmed last year. We became official molecular lab for confirmation. And we worked in collaboration with Tennessee Department of Agriculture. So Nate and Caddy would send us samples. They're from Department of Ag, pathologists, and the Forest Service people. So at the very beginning, we had issues with getting not so good samples. So we decided to modify the existing protocol and simplify it for foresters and people that are basically boots on the ground to collect these samples. What I want to mention here is basically the several counties, 6 counties, 5 counties basically near the Kentucky border is where the confirmation actually occurred. And there were 3 counties in Kentucky as well. But what is really alarming is the one that's in a blue in a very eastern part of the state. That's a Hamlin County. And that is near the Great Smoky Mountains National Park. And that got our attention because, and also did not mention that this is only on sassafras. So in Tennessee, we dealt only with sassafras samples. In collaboration with Romina and Romina's group, we are working basically on molecular detection. That is the part of the research. And I think Romina can say a little bit more about that in her presentation. I actually did not communicate with her regarding that. And as far as the beetles go, I'll let Bill maybe pitch in his part we did not detect beetles in the state of Tennessee. Bill, do you mind saying a word or two about that?
Just to say, the only work that was done with that was rearing from dead and dying trees, and I found Xylosandrus crassiusculus, Germanus nestus mutilatus, and Xylobarinus saxosenii, but associated with the sassafras. We haven't done any testing or trapping with eucalyptol or other volatiles to see if glabratus is present, but we're not sure where that would have come from. But it seems like other beetles are probably more relevant for vectoring. And why the jump to that Hamblin County, East Tennessee site, we're not quite sure what that is unless it's human-mediated transfer is a good guess at this point.
So one of the issues in our part, and this is, by the way, Bill, he's on an entomology side of the equation. I'm on the molecular plant pathology side. We received not so great samples at the very beginning, so it was a big learning curve for all of us because I worked with thousand canker disease with Bill in the past. So there was a quick learning curve for us to do molecular detection. And another part of the problem, samples started getting in large quantities. We don't have any funding to do this. So what we decided with the State Department is basically that they can send us a sample if we confirm the county. That is basically something that we're interested in. We'll call that county positive, and that's as far as we go for the state of Tennessee. And that is all we have. I'd be happy to take any questions.
Okay, I'm Yuli from University of Florida. I'm the moderator for your presentation. Thank you, Daniela. So quick questions from Matt Carson. He asked about, are you looking at northern spice bush in around disease sassafras? Did you find it in the field?
No, we personally not looking at anything samples. And so far, all the samples that were sent were from sassafras. That doesn't mean it's not there. It's just we're not in the field.
Okay, one more question from Academia. Was the CHK PCR performed directly on wood sample or on the fungal cultures?
So far, we had a pure culture and pure DNA. So that is something that we're playing with. We're very successful with Geosmithia morbida and direct drilling into the wood. And that is something we would like to explore. But I think several people are working on that. And that is something I'm interested in talking and potentially collaborating. But no, on this particular one, we did not. We just worked with the pure culture.
Okay, thank you. And probably one last question. Did you have see any fungi similar like positive in your test, but you are not really can confirm the identified?
So we had additional fungi that we sent to Ramina's lab, and maybe she can talk about that part. So that we got from the infested wood. And this is very similar to the work on microbiota that she published when she was with us. And I think her postdoc is currently working on that. But again, that is work from her lab, and I'll let her say a few words about that.
I think Jason means if you have seen any tree that shows symptoms, but you couldn't confirm that they had Rafaela lauricola present.
Yeah, in other words, did you get any that looked like you got something that looked a little bit like, or even was a Rafaela ultimately, but you couldn't confirm that it was lauricola, but they looked like it should have been positive, otherwise.
Yeah, so far all the samples that we received, we were able to confirm and they were positive. So we would not have any issues with that.
There's some in Indiana that there's something else going on. That's why I asked.
Interesting.
Yeah.
No, I was not aware of that. That's really interesting.
We can talk about it some other time.
So we have seen in Florida avocados with branch dieback, but caused by other fungi, not even Ophistomateles. So, sometimes you can get confused on what to expect and what you really
Yeah, I understand.
And sassafras is also susceptible to verticillium wilt in more kind of mid-Atlantic area. So it's possible that, just some natural low occurrence verticillium wilt could be mistaken for laurel wilt. I That's something that'll be teased out, I'm sure.
So one of the things we started doing, because this is relatively new pathogen for us, we created a metadata Google spreadsheet where we have all the information, and I started requesting pictures as well. So that helps, and all the symptoms look like a typical symptom. So there was really, which is a little bit refreshing to work with.
Well, there's a different Rafflesia, an undescribed Rafflesia that we recovered from Indiana, that in multiple places that we're working with. That's why I asked to see if you might have gotten it from Kentucky or Tennessee, and it is very, very, very closely related to Luricola. It's sort of like the aguacate story. And we're sort of working right now to determine whether it's pathogenic or not, or if it was the causal agent or just something secondary. It could maybe, anyway, I was just curious to see if maybe you guys have gotten the same thing.
Are you able to distinguish between the two then?
Yeah, ultimately, but it was, at first it took some work to figure it out, and some phylogenetic work. Tyler's was working on it and did the sequencing work and stuff. And it was just one that was not known before, and it appears to be a new species. But yeah, it was just one of those situations where it threw us off initially.
Thank you. So, thanks everyone. Appreciate it. Thank you, Danita.
No problem.
And Robin, you're on call.
Okay, let us do this. Okay, are you able to see this?
Absolutely.
Wonderful, thank you. So this will absolutely adhere to 3 minutes because I'm going to be basically presenting really initial stuff. So I was asked to talk about the current status of Laurel Wilt in Texas. My name is Robin Chaudhry. I'm currently at University of Texas, Rio Grande Valley. That's way down at the pointy end of Texas. Up until about last year, I was working with Karen Garrett at University of Florida. And I'll talk like 5 seconds about that at the end. Good God, come on here, folks. So Texas, most of the outbreak in Texas is occurring in the eastern kind of pine woods. That's up in this sort of region that just borders on Louisiana. And so I'm currently way down here. So I don't interact too much with Laurel Wilt on a day-to-day basis. But I wanted to talk a little bit about that. So, and then this map is the occurrences of Laurel Wilt based off of the dates. Danita just talked about this outbreak. I wanted to, and so this was the USDA Forest Inventory Analysis data set. And so basically this is the data set from USDA that just kind of keeps track of where different Luraceae are throughout the US. And so what we see is that most of the southeastern US is is red bay, then sassafras, and then over here is the California, it's the Umbellularia californica. So what you're not seeing is that there actually are Persea species as well as some other species, including Cassiathylia, I'm not even gonna pronounce that one, all throughout the Barrier Islands on the eastern side of Texas. So there are native Luraceae in these Barrier Islands, to my knowledge, and I've been through these Barrier Islands. I have not seen any Laurel Wilt or things that look like Laurel Wilt. That being said, I'm about a 5-hour drive from most of these islands, and so I've not really explored them too well. So right now, it's kind of a big mystery as to whether it's there at all. But currently Laurel Wilt is sitting around here, so it's possible that it could kind of hop down the coast. The next question that I always get after that is where would it go after that is Mexico. And so I just wanted to put in this slide of avocado production in Mexico. One of the largest reasons why some of us care about Laurel Wilt was for the agricultural side of things, avocado being the largest agricultural product. And so most of Mexican production is based out of Michoacan, although Tamaulipas and Nuevo Leon do have avocado production. There's a small amount of avocado production in the lower Rio Grande Valley, which is kind of where I live, but it's very, very, very small. So not a ton of avocado production down here. Most of what we grow is lula. If you all are familiar with that used to be really popular in Florida, and then it kind of went away for various reasons, including Laurel Wilt, among other reasons. And then I wanted to, and so the short answer is, what is the status of Texas Laurel Wilt? We know that it's in the eastern pine woods, but we're still trying to figure out whether it's in these barrier islands. I want to put in a plug. So up until about, I think like 7 months ago, I was working with Dr. Karen Garrett, who's also on this call. And so we were working on mapping for predicting out disease for the Florida avocado production and thinking about how we can use impact network analysis to model how disease might spread, both taking into account different sorts of control strategies. So this is the avocado production region of Florida with different sorts of groves sized out based off of their size, and then the colors based off of how many connections they have. And so we are still currently actively doing some of this modeling work, and we are hoping to get that out soon. And so that is it. I think that I beat the time limit. I want to say I beat the time limit. Anyway, onwards and upwards.
Okay, thank you very much, Robin. I'll start the 3 minutes now for questions. We've already got a couple that have come in. First of all, Yuri asked, who or which agency is monitoring for Laurel Wilt in Texas? It looks like Jason said it's Shane Harrington with Texas Forest Service.
Yeah.
So can you answer the how actively part of that?
So I can't answer the how actively. I have not coordinated very closely with Shane yet. And so I have not yet coordinated with Shane yet.
Okay, we have another question from Andrew Johnson. Zaloboris glabratus has also been found on other hosts, such as oaks. Are there other ambrosia beetles found in irrigated urban settings in the south area of Texas? So are you dealing with other problematic ambrosia beetles in your area?
We know that there are ambrosia beetles. That being said, I am trained as a plant pathologist, and I have not been taking too close of a look at the entomology side of things. That being said, I am open to suggestions. I am open to suggestions and collaborations, but I am not a trained entomologist, so I don't really know too much about that.
Okay, so I noticed on the Lauraceae map that there's quite a big gap along the Mississippi River. I assume where Lauraceae don't work so well. So how do you reckon it crossed that gap? Was it human, or is that, you know, I see there's a couple patches in there.
So human-mediated transmission is probably one of the best transmission modes. And so we know that from basically, if we look back at this, we know that there are several outbreaks that occurred extremely early in the epidemic. And so those early outbreaks would be, my guess, would be trade, would be where people may have accidentally moved it through firewood, or transmission through maybe logging wood, like pulpwood. I know that some of the early outbreaks in this region were associated with like some of the longer but like not super long transmissions were associated with pulpwood through rail lines.
Yeah, it looks like it kind of popped up near Shreveport up there really early on, and then off west of Lake Charles. So is that associated with timber mills in some way or something? Oh wait, so we have a question. Just so we don't run out of time, Jason asked, has sassafras been found to be impacted in Texas?
So I don't know about sassafras in Texas. That's a really wonderful question. It does exist in the eastern pine woods, but I don't explore nearly enough, and so that is something that I need to coordinate more closely with Shane with.
Thank you, folks. Appreciate it.
Thank you very much.
Great.
Thank you, Chase. Thank you, Robin. Let's move on to Andres. Welcome to our conference.
Hi there. Thank you for the opportunity. Can everybody listen well?
Absolutely, we hear you. And if you can tell us which institution you're with, that would be great.
Yeah, I'm from the Institute of Ecology in Mexico. This is in Jalapa, Veracruz, the Gulf Coast part of the country. So I've been working with ecological niche modeling and more on a macroecological perspective to understand, what are the potential distributions of these beetles and the associated pathogens that they carry. So we use this framework that you see on the screen, which is basically combining the abiotic, biotic, and movement factors that can then explain the distribution of a species, what we call the GO part, which is
we don't see your screen.
Anders, can you push the button share screen at the bottom?
Oh yeah, sure. Share. Okay. Can everybody see there?
Not yet.
Okay, sorry.
You may need to choose the right screen.
Desktop, yeah, right. Compartir. See if it's working there or not. Okay, sorry. Share. Okay. Can you see my screen now or not?
Not quite. If
there.
Oh yeah, it's coming.
Sorry.
Yes, thank you.
Sorry for that. I didn't have activated the.
Yes, we see it now.
Okay, thanks. Can you see it there? Yeah.
Yes.
Okay, so I was referring to these three sets of factors, abiotic, biotic, and movement, that we then would like to find in the field. So if they intersect, then we would be able to predict the distribution of the species, or what are the potential areas for this species. So what I've been working with some students and colleagues is to understand this from different perspectives, trying to bring together what we call different dimensions of biodiversity, basically working on geographic space, ecological space, and now we have been moving on the phylogenetic space to try to find what are potential hosts and that may cause risk. So if you see, there are different things I'm going to show you. On the left part of this slide is the summary of what we've done for a correlative approach. Basically, this is using abiotic and habitat-related layers using ecological niche modeling based on the occurrence of the species, different species it may well be, where the hosts are or which hosts are these, or the beetles themselves. And then we can project the potential distribution of these beetles or plants on geographic space. And that's what we see here. We see where are the more suitable areas for the establishment of the beetles given the presence of hosts and abiotic and biotic conditions. So in the bottom part, you see the map, which corresponds to Helleborus glabratus potential distribution in Mexico. You can see there is a broad area, and that's because there are a lot of hosts in Mexico that may be suitable for the species, given what we know. There is a little four set of maps here, and it's a summary of where are the conditions for ambrosia beetles as well as for lauraceous species. And in the bottom part here, you can see the richness maps, which is where these species concentrate. So you can see the hot spots of biodiversity in Mexico. And before, Robin was already mentioning that Mexico is a major producer of avocado, and that concentrates in the central part of the country. And so with these maps, we could probably extract which are the municipalities that may be more vulnerable and that may be of high concern. So if you move then to the right part of this slide, you can see what we've done in terms of what we call process-oriented modeling, which is not anymore like a static perspective, but is based on the demography of the species and what we know about the rates of dispersal, given what we find in papers or different data we can use. So we can have different scenarios for the species depending on where we set the seed for the model to run. So in the bottom right part, you see the prediction for Helleborus glabratus. If we let it start from Georgia, and then we start the simulation, and we see in fixed lines, the black lines represent what are the suitable areas for the species. And on top of it, we let the model run, and you see yellow to red, a map, which is the predicted abundance for the species given the right abiotic conditions. So you see, it finds a good spread of the beetle appears on the east part. And then when it finds the right conditions in central Mexico, it quickly starts reproducing and settling there. So that's why you see that big jump in central Mexico. And that's because of the right conditions after the beetle arrives. That's what the prediction says. So in the upper part of this map that I just showed you, I'm showing you what we're doing right now in terms of having more accurate demography for at least 2 beetle species, which is Xyloborus bispinatus and Xyloborus affinis, as a function of temperature, because we want to have these dynamic modeling predictions for what we could expect for climate change. So the idea is to use these data, which we need for nurturing these models, which is basically demography and dispersal rates. In this slide, what you see is another approach that we have been using, which is using phylogenetic relationships of hosts and the occurrence of different vectors associated to these hosts, to then trying to find where are these potential hotspots of interaction, either worldwide or in different regions, depending on which data you use. So what you see here on the upper part and right part of the slide is 2 examples of different beetles that have different, have been related to different hosts. So for example, Xyloborus glabratus is more like a narrow host range because it's more associated, at least the pathogen that it carries and the interaction of this beetle is more associated to Lauraceae species, whereas Eualace fornicatus is a different story. This is more a widespread and a wide host range beetle. So then using logistic regressions, borrowed this idea from Greg Gilbert, we then can predict what you see in the graphs here on geographic space, and that's the little map you see there. The brightest areas correspond to where you would expect to find more concentration of potential interactions of these beetles with different hosts, given the phylogenetic distance of these hosts. So you can see we can replicate well the Asian part where these beetles can come from, then some perspective on Europe where we know there are hosts that are potential for these beetle species, and then of course Mexico and Central America, as well as the eastern part of the US, which is where we know the beetle has already been established. And of course, this is at very coarse resolution using coarse climatic variables, but at least I think it can be useful for finding places where we could go and do some surveys and other interesting experiments in the field, or at least try to predict other hosts that we yet don't know where they are. And with that, well, this is another zoom to the area where you already have shown, Robin. And the model we are using, as you can see, predicts very well on time where the municipalities or counties have been already affected, and some other ideas for what the barriers, natural barriers are, which is using NDVI analysis. And with that, I would like to thank you.
Thank you, Andres. Great. Lots of work. We appreciate you being here. There is one question from Jason Smith. His lab has tested susceptibility of a few Lauraceae native to Mexico, such as Persea podadenia and others, which is very susceptible. Would those data help you improve your models, or?
Yeah, I think so. They would all the presence data on all confirmations can be very valuable for this type of modeling. Either if you use them to start a model, or then to predict if it predicts accurately, you know.
Great. Then you guys get in touch. That would be great.
Yeah.
One other question is, what have you tried to project your models down to South America? I think there was one map where you have shown that, yes, the last one, that glabratus is comfortably will be comfortable in South America, right?
So that is what we've done. The map you see here, but we haven't done yet extended the dynamic or process-oriented model to this part. I know there are other guys working with avocado in Colombia and other regions that have started working with this, but this is easy to do, you know. So it would be easy to have a prediction for this part of the world.
Awesome. Thank you very much. We are moving on to Jason Smith. Jason, are you with us?
Yeah. I need to share my screen. There we go. There we go. Now, let me get my screen up. There we go. I think that's it. There we go. Oh, sorry about that. Takes me a second. Okay. Is that working? All right. So,
see the presentation.
so I'm going to try to summarize. I'm trying to summarize about 10 years worth of work, and also a couple of graduate students' research here as quickly as possible. So basically, we set out to try to identify potential resistance in native hosts, and this work began in really it started in 2008, and the basic concept was with the very, very high mortality rates that were essentially widespread in both Red Bay and other hosts like Swamp Bay. The concept was that we could go out and look for survivors, and the hypothesis was that those survivors would be a good source for tolerance, and we could test that by propagating them and look to identify potential resistance mechanisms, and then go on to do breeding work or potentially see if that resistance would be heritable. So this work began with Mark Hughes, a PhD student in my lab. He began by searching for these tolerant individuals, potentially tolerant individuals in some of the most affected areas in coastal Georgia, South Carolina, and Florida. These are areas where you saw essentially 90 to 95% mortality in stands within the first 3 years. And he did a survey for these trees, propagated them, came up with a propagation method, which there were no published methods at the time. This work was published in the Native Plants Journal in 2014, so you can look that up if you'd like to. He at that time, initially there were 60 genotypes that were propagated. Over time, we have been screening and verifying the resistance in those trees, and a number of genotypes have indeed been verified that are tolerant. And in addition to that, we've been working with other colleagues like Kathy Smith with the U.S. Forest Service, and have identified that basically one of the things we do is verify essentially the distinct nature of Red Bay versus Swamp Bay and Silk Bay. So there's been some disagreement among botanists in terms of the differences. The importance of that is to understand how we might go about restoration. And so we've done some genetic work, and also developing some genetic resources like microsatellites and things like that can be used. And so I just wanted to point that out. But one of the other things that we've been doing is looking at mechanisms of resistance within some of this material. And we've worked with some collaborators at the University of Minnesota in Bob Blanchett's lab to look at the difference. One of the hypotheses has been that the tolerant trees may either perhaps they may induce resistance mechanisms very, very early on, or perhaps they actually don't react at all. And so we've been looking in the vessels and anatomically using electron microscopy. So that's one of the things that's been happening. In addition, we've also utilized tools like RNA-seq, gas chromatography, mass spec, and other things. And again, utilizing this clonal material. And then more recently, I had a master's student, Matthew Eichholz, who just recently completed his thesis work. And he has gone back and reevaluated this material that was initially identified in 2008, and has essentially looked at the survivorship of this material. And you know, some of them have died, but a lot of these trees continue to survive. And again, this is in regions where we normally see 90 to 95% mortality. But what's very interesting is that he's been looking at recruitment around these trees, and what we see is that we're seeing essentially regeneration, and there's definitely a difference in the growth of the regeneration around the survivor trees versus plots that were set up in 2008 around dead trees. So we actually have this clear natural regeneration that's occurring on the sites that had the survivor trees as sort of a center of the plot. And so that provides us with some hope that these survivor trees are actually contributing to the natural regeneration, and perhaps there is some tolerance that is somewhat potentially heritable. So we are actually now testing the heritability we have seedling populations that have been planted out in the field, and we're in the process of testing that material. We're working with our one of the new hires at UF here that is looking at genetic resources and resistance. And so we're going to be testing that. Jeremy Bronner, who's in the plant pathology department, is going to help with that. And then lastly, one of the things I wanted to point out that we've done, which I think is an interesting thing, is we have done some experiments. One of my recent postdocs did some experiments where we inoculated leaves. One of the things that we found early on was that we could recover the pathogen from leaves after we inoculated the stems. And so we wanted to ask the question of whether or not we could inoculate leaves and get systemic wilt. And so here is my postdoc, June Lee, inoculating the leaves of a silk bay tree. And the same tree, basically 2 months later, has systemically wilted completely. This is using a hypodermic needle. Here's just a photo showing basically cultures that were established from the stems of the same tree. I'll plate it out, and then you can also see we've also basically shown you that you can isolate the pathogen both from leaves and stems. Here's another photo I just wanted to show you real quickly. In the greenhouse, we did a lot of different experiments where we inoculated leaves using different methods, both scissors, needles. We also used carborundum as well. But we did this with avocado. And the point is that I just wanted to mention that we have a hypothesis that there may be other transmission methods in addition to ambrosia beetles, that it may very well be possible that leaf-feeding insects should be, we think they should be considered as potential vectors. The pathogen is very, very small. It's essentially the size of bacterial spores. And so it may very well be possible that leaf-feeding insects could be another, possible transmission vector. So with that, I'll end it. And thank you very much.
Thank you, Jason, for the presentation. I'm having here a couple of questions. Matt was asking if the all the resistance trial were done in orchards, or were you out planting genotypes in disease epicenters?
Okay, so I'm sorry, what was the question one more time?
If the resistance trial were done in orchards, or were you out planting in disease epicenters?
Oh, sure. So there is natural occurrence in the where the trials were done, but we artificially inoculate. So, but we've also done some restoration plantings now as well. So we do have some trees planted out in natural forests where we've done some restoration experiments as well. So those will be, evaluated over time in addition.
And Benita was asking if what was the inoculation method that worked the best when you were trying to inoculate leaves?
Oh, actually, so it was actually scissors that worked well, believe it or not, where they were just dipped with inoculum, worked really, really well. The hypodermic needle, it was sort of hit or miss. It doesn't work all the time. So again, it's probably something that's infrequent. We're not suggesting that this is the primary, obviously not the primary method. The point is that it may occur occasionally, and it may explain some of the transmission that may be occurring that we can't necessarily explain otherwise. Possibly, perhaps in the avocado groves where we're seeing transmission and we don't understand why it's occurring otherwise. And so I guess that's the point that we're trying to make here.
All right, so I have 3 more questions and then we'll have to move on because we're a little bit back behind. So Robin was asking if the leaf accumulation, the reason for reduced abscission, or is the change of hyperspectral imaging?
Yes, so it's possible, but not necessarily, because, you know, as the plants start to wilt, there's obviously other biochemical changes that would be possible for, you know, explanations for that. We don't know why that happens, but we start recovering the pathogen in leaves in about 10 to 12 days, something like that. But we know that there's biochemical changes that occur before that. Within 3 days from some of the work that we've done with other colleagues. In fact, you'll actually Xavier Martini has done a lot of work on this with some of the biochemical work. Within 3 days, you get to some of the biochemical changes, which would explain that. So, yeah, that.
Yeah, also ask if there's something like constitutive that might be involved as well.
Right.
So Ross was saying that the Rollins suggested the Raffaello rollicola genome may contain genes coding for the effectors that can elicit host defenses. And do you think that tolerant species might have genetic differences related to the defense responses, and comparative genetic study between tolerant and susceptible cultivars might uncover these differences? Jason? Did Jason disappeared from the...
He did.
All right, well, I guess he will not answer to the remaining question then.
Okay.
You can always, I mean, we're gonna have all these questions available, so we can always answer later on through email and follow up.
Thank you, everyone. Let's do a mandatory 3-minute break. So we'll get back at the 55 after the hour, whatever time zone you're at, just to stretch our legs, and then we'll have to move on.
Jason just said his computer died.
Oh.
Okay. We'll see you in 2 minutes.
All right. I will already start sharing the screen so that once we're back, we're ready on mine and ready to start.
Thank you, Katarina.
You're welcome.
I think we can get started again.
All right. I hope everybody was in time to get back from the break in a few minutes.
Welcome, everyone.
Gary, let me know whenever I can start.
Yeah, I see 86 participants. I think we can get started.
All right. Well, hello, everybody. I'm Caterina Villari from the University of Georgia, the Rollins School of Forestry and Natural Resources. And the work I'm presenting today, it's actually the thesis of my master's student, Jeff Hamilton, here in the picture, and who just graduated this semester and who, by the way, is searching for a PhD. So I'll just leave the information there for all of you. All right. So, so far, as even Danita was mentioning, when you find a sample and you want to confirm if you have Laurel Wilt or not, right now you probably have to ship it to some certified laboratory where they're going to have to plate the sample, isolate mycelia, extract DNA, and then do PCR work, sometimes even sequencing. So it's easily that you get up to 9 days, 6-9 days before you have a confirmation. And that is definitely too long, especially if you want to do rapid response and take off the plants right away, or you have to intervene quickly. So we wanted to propose an alternative way to go. And in particular, our proposed option is the use of LAMP, which stands for loop-mediated isothermal amplification, which is a DNA analysis amplification method. So basically, it is similar to PCR, but has the feature of being completely field portable. So you can do it directly at the point of care, and it's very rapid. You're going to have a response in less than 90 minutes. So if you compare 90 minutes with the 9 days that you would need if you send the sample outside in a lab, of course, I mean, you could see the advantage. But this is the final product. How do you get there? Well, first of all, we designed the assay, and we designed it to be specific on the beta-tubulin gene. And we verified it was highly specific. It doesn't cross-react with any other species. And it's also very sensitive. It amplifies as low as 0.5 picograms of DNA, and also as few as 50 conidia, which is more biologically relevant. I mean, the insect will carry conidia, not picograms of pure DNA. So we tested with conidia, and as low as 50 conidia, it's capable of amplifying it. We also want to know how early in the disease cycle we could actually detect the pathogen. So we did a time course experiment inoculating plants, and we saw that, first of all, the assay works perfectly, both with host tissues, no need to isolate the mycelia, and insect vectors. So very useful for surveying, for instance, and it works perfectly with both pure DNA and crude DNA, which is crucial if you need to go in the field. And you can detect it as soon as 12-day post-inoculation, up to 50 centimeters above the inoculation point. So moving to the field, we wanted to see if we could detect it in field collected samples across 6 different southern states. But the final validation was really going into the field. And Jeff literally put everything in the back of his truck and drove to 5 different states and validated the assay in field condition, in the condition that you can have when you go out and survey. And he confirmed that we can have an accurate diagnosis in less than 90 minutes. No matter the weather condition, wherever you are, you can have confirmation. And once we validated the result in the lab, we were able to see that the sensitivity, so the true positive rate, it's higher than 90%. So really good for a field test that give you a result in 90 minutes compared to the 9 days that you need. So take home message, it works. It's pending publication. We're minor review pending, so it's going to be out there soon. We are ready to train people, and it's working. So just please use it. It's for your own convenience. And I'll leave here all the acknowledgement, especially the funding agency, while I'm happy to take questions.
Thank you, Catalina. So you have a bunch of questions right now. So how many other species, reference species you had been tested, and other closely related fungi also you found? Because we know so other referrals can be also associated with beetles when they attack the trees.
So we tested almost 29 different isolates of Rafaela species and other closely related species, Umbrella species, other Ophiostoma species, and so far none of them cross-reacted, including the aguacate that was recently discussed during this meeting. So so far the ones that we tested, most came actually from your lab and David Carrillo lab. Harrington's lab, we receive isolates from all across the US and none of them cross-reacted. So
thank you. And did you ever test it on the new Raffalius Jason just mentioned they recovered from Indiana?
Not the one from Indiana. We didn't receive that. So we would actually be curious, but the one from, from Indiana, not being published and not being available, we were not even aware before this meeting actually. So we'd be happy to test it, but.
Thank you. And one last question, I think from Matt Carson, how cheap the equipment?
So the equipment is it's relatively cheap if you think. So those the portable instrument over here, and there are different brands available. That equipment there is $8,000. So it's not that much. And once you have it, you could bring it with you in the field. So it's not probably something that a private person can buy, but we're training the Forest Service to have it. And once they have one equipment, they can use it for many different pathogens. So it is actually a good investment. With $8,000, you have one thing that you could bring all across the state, and it works with different primers for different pathogens. So it is actually affordable, way more affordable than thinking of sending stuff to external labs, if you think of the advantage in time.
Thank you.
Or like a port of entry, you could use it at a harbor to screen material. So it is affordable.
Okay, thank you. Thank you, everyone. We have so many questions, but we have to keep it to 3 questions of each presenter.
I'll go screen them and perhaps try to respond privately if I can.
Thank you. That would be great. That's a very good option. Thanks, everyone. Let's do some of the questions in the chat or later. People can reach out to any speaker later. I would like to welcome D. Mills and Julian Mendel, I think. Is that correct?
Yes, I'm going to have Julian do the presentation. My provost just tapped me yesterday on a project that I'm going to have to jump off here, so I want Julian to give the talk.
Hi, everyone.
We are from Florida International University. Sorry, Julian, go ahead.
That's all right. Hi, everyone. My name is Julian, as Dee said. So I wanted to talk to you guys about some detection work that we did that is not molecular based. It's using canines, as you can see. Our research has been published in 2 publications in horticultural technology, and they're there for you if you want to read more. I'm happy to send them to you as well. But our research, we kind of separated these publications based on training the dogs and determining the accuracy with which they can detect Laurel Wilt disease, and also in deployment, where we actually worked with grove owners and farmers in Homestead in the Redland area, and were able to evaluate it as a tool for early detection and successful treatment for containing and mitigating the spread of disease. So very briefly, and I'm happy to take questions to clarify anything at the end, of course, but we utilized a patented material called COMS, controlled odor mimic permeation devices. And essentially it's a polymer bag that has a fixed pore size that allows odors to be sealed inside and permeate to the outside environment at a constant rate. We also tested this to ensure that spores could not pass through the bag in plating experiments to ensure we weren't accidentally spreading Laurel Wilt anywhere, because we did work in the field. We did this study of training for about a year and a half. We did 229 training runs with 3 canines. And we did this in mango groves instead of avocado, because we wanted it to be as controlled as possible to not risk coming across an actually infected tree. So as you can see here, we were able to demonstrate that the canines were able to pick up Laurel Wilt disease infected wood in avocado wood with very high accuracies and positive predictive values. Each dog had a different rate, but in each case, 90% and above accuracy was determined with these dogs. So once we completed our training process, we went into deployment and we worked with about 11 grove owners. 9 of them we worked with on an on and off basis, but 3 in particular I wanted to highlight that worked with us consistently. And we essentially went in through the entire grove and ran the dogs, and we would detect trees, tag them, GPS coordinates, and if the trees were detected by 2 dogs of the 3, we would indicate to the grove owner that we recommend they follow the treatment protocols with tilt, 2 or 3 rows of trees surrounding that indicated tree. And you can see in this table the number of trees in the grove, the number of trees we detected, how much they treated, and then how many they lost to wilt within a protection period, as you can see that we observed. So in particular, Grove 1, I wanted to mention, had lost 100 trees in the year before we started to deploy dogs in their grove. We found 35 trees, pre-symptomatic stage, so no visible wilt. And then we actually recommended our treatment protocols. And only after the 12-month protection period of tilt that it is recommended, we actually saw 4 of those trees that were indicated progress to wilt. So they were a little bit slow on reinfusing with tilt in that case, but you can see that it's a pretty successful deployment tool, early detection tool that we were very pleased with. I wanted to also highlight on the bottom right there, you see an area. So this is an avocado grove that was about to be bulldozed, taken down. The grove owner had given up on this grove, but they allowed us to go in with the dogs and do an observational study for 6 weeks. We wanted to see, we let the dogs hit the trees and just see what happened. So we weren't treating, we're observing to see how long they progressed to wilt. And one of the interesting findings we found was that up to about 40 days before symptoms appear, we were able to see that the dogs could detect it pre-symptomatic stage. So we have a decent window of detection there before the tree goes into full-blown wilt. So I wanted to talk about, before I get into the metabolomic slide, very briefly, one of the things we're also working on is looking at the volatile organic compounds using GC mass spec that are associated with Laurel wilt and Rafaela lauricola. And we are actually doing a new study working with Hawaii Forest Service targeting a new disease called rapidohea death that has very similar characteristics to Laurel wilt. So we're actually looking at comparing those volatiles to see if there's anything similar between the two diseases. And the last part of my dissertation involved metabolomics, where we were looking to see what biochemical changes were occurring during disease from early to late stages, as well as comparing healthy to infected xylem, and as well as gallery flushes, where we would actually use sterile water and flush beetle galleries out to collect and flash freeze and go to LCMS. So during this study, we actually detected 171 metabolites that were differentially up or down regulated. 104 of those were significantly upregulated in infected xylem, 11 were found exclusively in infected xylem, and 10 were significantly downregulated in xylem. I wanted to point out that with respect to the detection world, I know that the DNA analysis, a lot of times, especially at the early stage, the distribution of the fungal spores is not necessarily uniform and it's very patchy. So a lot of times we get these false negatives. But we also utilized machine learning algorithms with some of this data to determine whether or not we could use these algorithms and the metabolic profiles to classify and determine if a tree is infected or not. And we were able to get accuracies using this data. Of course, it's a limited number of samples, but with 98% when comparing infected wood to healthy wood, we're able to classify them correctly. And then we were also comparing gallery flushes as a suitable sample to infected xylem, and we're able to also classify with over 90% accuracy using random forest algorithms. So this is another way using non-DNA tools to actually detect and identify diseases. With that, there's a lot more to talk about, but I'll happily stop and take questions if you have any.
All right, thank you, Julian. We have a couple questions. The first was, did you treat only trees next to pre-symptomatic ones?
So our guidelines kind of followed the guidelines that were coming out of TREC, which was that if a tree was detected as symptomatic, you would recommend to remove that tree completely and treat about 2 or 3 rings of trees surrounding that, I believe. So in some cases, we didn't actually determine what the grove owners did. We worked with them, we gave the recommendations, but in the cases of those 3 in particular, they would treat not just the pre-symptomatic tree, but also the tree, 1 ring of trees surrounding that at least, is what was done in those groves.
Another question from Janita is, who takes the dogs home? Who basically manages the dogs when they're not in the groves?
That's an excellent question. So we actually, once our grant was up, we created a spin-off company that called Innovative Detection Concepts, and that company, they live at Dr. Mill's house, believe it or not. So they're when they're not working, they're pets, they're lovable, they're just like any dog. They're not just you know, to us, a working tool. So the dogs go through a constant program of enrichment and constant refresher trainings. We've also trained them on additional pests, additional problems, including invasive lizards, tegu lizards, and pythons and things like that. So they keep up to date with their training, and they're also housed with this company that manage them, and we do deploy for different diseases and pests as well.
Okay, in trying to keep with the time, because you went a little over, and that's okay. But there's some questions for you in the Zoom group chat that I'd like you to address, but we need to move on for keeping on track.
Okay.
Thank you for your time.
Thank you.
Pedro is next, and Pedro will be talking about culture-independent Laurel Wilt diagnostics. Take it away, Pedro. Pedro, I think you're muted if you're talking, because I can't hear you.
Now, can you hear me?
Yes.
Okay, I'm sorry about that. Good morning, everyone. My name is Pedro Pablo. I'm a part of the Laurel Wilt Research and Extension Program at the University of Florida. I work at the Plant Diagnostic Clinic, and I'm gonna talk to you about detection, especially in avocado, but also we are working on some other Lauraceous species. So in the left-hand side of the slide, you will be able to see a figure, a graph with the general model of how Laurel Wilt spreads within the avocado orchard. So first we have beetle inoculation, and after that, another way of transmission of the pathogen is through root interaction of trees, right? So it's a very important way in which Rafaela auriculata spreads. You can also see in the pictures a planting, a tree that has been heavily affected by the pathogen, and we have been losing trees since 2017 when the first outbreak occurs. We have removed over 65 trees out of 150. On the right-hand side, you can see an animation of how we have been progressively removing trees and in this very planting. So root interaction happens, and in Florida, it's very common, and there are several factors that can influence that because the high density, because the water table, because of bedrock, and several, and of course the age of the planting. So we cannot prevent, in many of the cases, the roots to be in contact with each other. So in order to prevent the spreading of the pathogen, a timely diagnosis is crucial. So for having a timely diagnosis, we need it to be, of course, rapid, early, but also it needs to be reliable. If we want a methodology to be reliable, we need that to be specific, sensitive, and the level of agreement among the replicates to be consistent. When it comes to specificity, we need to avoid false negatives, and we need to avoid false negatives and false positives. So we have tested 101 strains of Rafaela auriculata isolated from different hosts and different origins using IFW and CHK that have been already developed, and we have had 100% of amplification rate using DNA from pure cultures. In order to avoid the false positive, we have assessed these primers or screened these primers against other Rafaela species and non-Rafaela species that have been isolated from Laurel Wilt infected or Rafaela auriculata infected trees, as well as 101 endophytes isolated from healthy avocado trees. So in total, we have screened over 280 strains of different fungi associated to both Rafaela loricola infected trees and healthy avocado trees. Also, we have discarded false positives by screening 11 different avocado cultivars. We also found at the end that IFW, when we talk about repeatability and consistency, IFW seems to be more consistent than CHK, which results were more variable. As I say, in total, we have tested over 280 different strains in 90 species. So now that we are sure that we don't have false negative, false positives, we moved into direct detection of Rafaela loricola from sapwood. So we assessed 85 sapwood samples that were previously confirmed with Rafaela loricola, and we use IFW and CHK. As I said, IFW is a more suitable marker, and we found a detection rate of Rafaela loricola of 89% when we use IFW. Contrastingly, we found that only 9%, around 9% of detection rate when we use CHK. So we optimize the whole protocol for IFW to be performed in only 24 hours. So what's next? What's in progress? So we need to further adapt this methodology, which is ready to go, but we needed to further adapt it for an early and direct detection from roots, ideally from asymptomatic trees. So we have a better recommendation of how many trees we should remove in order to save more trees from being infected with Rafaela loricola. Of course, trap, capture ambrosia beetles, and for future use in sentinel programs. Something I would like to highlight is that we are ready to go. It's a technique that you can just perform in 24 hours, and it's set up for diagnostic clinics. You don't need any additional equipment that you already have. It's easy to perform, and no further training is needed. And of course, our fungal collection and the methodology we have optimized is the baseline to move forward to more sensitive techniques. And with that, I would like to thank the Forest Service for allowing us to use avocado as a model. Of course, the research project team, which is in conjunction with Genita, the University of Tennessee, and of course, the lab members. And with that, I'll be ready to take any question.
All right, thank you, Pedro. Katerina had a question, and she said in her study, they had issues with CHK cross-reacting with host tissues. Have you found similar results?
Yes, when we tested CHK, it did not produce any amplicon when we use DNA from 11 cultivars. We didn't get any cross amplification or any amplification from host tissue.
I have a kind of related question, and it looks like you tested a bit of diversity in and around Rafaela. Have you tested mixed template cultures and things like that? You know, thinking about more natural sampling or samples might contain multiple fungi and different tissues.
Yeah, not like a mock population or mix of DNA extracted from pure cultures, but we have that situation when we extract the DNA from sapwood samples that were infected, because in that infected tissue, there is not only Rafaela, but there is any sort of different fungi. So I would think that would be the case. So, and we have also sent for sequencing some of those PCR products that we obtain when we amplify IFW from these sapwood samples, and they correspond to the target region, which is the IFW microsatellite region.
Okay, great. Jason Smith says, using only SSR may be risky since it's always possible that it may amplify from something else. Two were originally chosen because it greatly decreases the chances of random amplification should a new marker be developed.
Well, so far, IFW has worked really well, and we have tested in, as I said, is 60 different genera and 90 different species that have been found in association. So if this primer is working, well, it's, I don't see, well, it would be very good to have another marker, but we can use this one for now until we are able to develop other ones. But I think we are, at least for now, we are good to go with IFW.
There are,
thank you, Pedro.
there are hundreds. There are hundreds of SSRs that one was created from. So there, we have them. If if you wanted to just make another one, it's not hard to do. I'm just saying, if you wanted to just
Okay.
So you could have two, and that would make it much stronger assay.
Okay.
Yeah. Thank you, folks.
Thank you very much.
I think we have to move on.
Awesome. Thank you, folks. We are entering the final, very exciting part, the management. How do we use all this knowledge in the field? And we're going to get started with Romina. Again, I'm sure you guys are all conscious of our time. We are going to go a little bit over 11:30. In theory, Zoom should let us do that quite easily without kicking us out. So let's see what happens. But thanks everyone for being conscious of time and looking forward to it. Romina.
Hey, thank you. Can everybody hear me? Yes?
Yes.
Am I mute? No, right? Okay. So thank you for sticking to the last section, which is management. And as Daniel mentioned, we are in the epicentric of avocado production, Laurel Wilt impact. And so we are always, we have the pressure of growers in a good way because they want answers yesterday, right? So, so I'm going to talk about some crazy ideas that we are bouncing among people at Trek and also with input from the growers. I'm from the University of Florida, from Trek, also part of this Laurel Wilt research and extension group. So this project or these different experiments came about because we made the observation that there are some surviving trees in the landscape. Landscape meaning avocados orchards. And this was actually first observed by growers, and we kept hearing about it, and I was really skeptical because we know how virulent and devastating is this disease. So I was surprised that there are some standing trees even being productive. So we went to the field and we are monitoring some of these surviving trees. This is not a single case. There's multiple cases of this happening in the landscape. And this is an example that you can see in the upper side of the slide. This is a tree that was diagnosed with Laurel Wilt in January 2019. And last week, we got more than a year after a second season of production of fruits. So you can see that the tree goes through a rough patch, right? But it seems to be still standing and producing. This is not, our recommendation is not to leave the trees that are infected on the ground, of course. But we have good collaborators that are avocado growers that are letting us study these trees to try to understand what's happening. So the first thing we did was to go to the field and collected samples from these surviving trees. And to make things more complicated, in Florida, there's hundreds of cultivars. And on top of that, people usually have 3 different cultivars in one tree, right? Not to make things more complicated than they need to. So in this case, the root stock was Walden, middle stock of Dupuy, and a scion of Eru. So we sample all these different heights using a core borer. We follow the traditional methods of diagnosis. We recover Rafaela lauricola. We confirm with the microsatellites and with LSU. So we are sure that this is lauricola. And furthermore, we isolated some strains from surviving trees, and we have inoculated trees, and the trees die. So these are virulent, just common garden lauricola species. So one thing that we observed that was kind of like a pattern is that these trees that we were finding surviving are adjacent to trees that were removed because of Laurel Wilt. And this is a common pattern. So the tree that I show you, the sequence is 9-7, which was adjacent to 2 trees that were removed. So then, you know, after a lot of discussion, we were thinking maybe these trees have been infected with the pathogen through root graft in a slowly manner, and somehow they learn to live with the pathogen for some reason, right? We don't know really the mechanisms behind it. And so again, because this disease is really being explored by many laboratories looking at the basic biology of the pathosystem, there's a lot of things to discover. So my lab has taken a backwards approach, right? We are going to try different things, see what sticks, and then try to understand what's the mechanism behind whatever it works. So we went to the avocado committee, Florida Avocado Committee, and proposed to try to recreate what we see in the field and try to test different vaccination, in quotes, treatments. They gave us money. So we were able to, this is a picture of my shade house. We were able to build a collection of avocado trees that are representative of the industry. And one thing that is important is that working with avocado trees is challenging because there's no many available right now because people are replanting. So we are kind of competing with growers in trying to get these small trees to do experiments. And so we propose to do different treatments to elicit the response prior to challenging these trees with the pathogen. Several treatments were proposed: low count Lauricola, deactivating inoculum, sterile filtrate from Lauricola, non-pathogenic Rafaela, and a product called Dutch trick, which is used for vaccinating elm trees against Dutch elm tree disease. And so I'm just going to show this experiment. We started with a low count. So we inoculated trees with 600 CFUs. And this was done in January 23rd. One thing to consider is that it's winter in Florida. Even though it's warm, there's still, you know, affects the evapotranspiration of the tree and might slow down the colonization of the fungus through the vascular system. So we inoculated January 23rd. This is a picture on February 2nd. We got worried because nothing was happening. So we harvest some tissue. We were able to detect the pathogen up the stem and recover the pathogen. So the pathogen was present. March 18, you can see what we call sadness or tristeza, which means that the plant becomes droopy. Now, these plants recover. And so we have this group of plants that were inoculated in January 23rd went through a period of tristeza, of sadness, just like we see in the field. But this picture was taken May 13, and you can see they're okay. So I couldn't wait. We are gonna repot them and let them grow, but I couldn't wait, and I re-inoculated one of these trees with 3,000 CFU, which is the average amount that we inoculated into experimental trees. And you can see that it again went through this sadness period, and then I took a picture 2 days ago, and it has recovered. What this mean, I don't know, right? So, but it's worth to keep looking into it. So we are going to scale up this experiment. Another experiment we are planning to do very soon is the inoculation of all these different Rafaelas that we are isolating from avocado, from healthy and diseased avocado trees that Pablo has been collecting. So once we show that they are not pathogenic, we are going to use them to see if we can elicit a mild response in the host, and then challenge them with the Lauricola inoculum. And so with that, we are looking at other things like testing different cultivars, testing different grafts numbers. So we want to stockpile different grafts and see if that how is that related to disease progress. And we are also going to look at root health, the status of the root health in terms of disease progress. So thank you to the Forest Service again for letting us use avocado as a model system, and to my research team and collaborators.
Thank you, Romina. We have some questions. Diana asks, have you searched for some molecular markers in the trees exposed to low doses?
No. So again, this is kind of, this project was born to really answer some of the growers' demands. And they are doing experiments by themselves in their groves. So we are just trying to give a more statistically sound context to those experiments and kind of recreate what they are doing and observing. But my plan is to stick to 1 or 2 treatments that show some promise and then go back, right? We can scale up in number of replicates, we can scale up in number of cultivars testing, and then resource to our collaborators in other laboratories that can look at the pathways and at the transcriptome and other things that could be happening within the host. But we need to narrow down the number of treatments as for now.
Thank you. Chase asks, could this explain the resistance in native populations in Asia that they are vaccinated?
That would be great, right? Of course, we don't want to, I have to be really careful when I say vaccination, especially with low count of Rafaela lauricola, right? I don't want in health to recommend to have trees inoculated with the pathogen more than we already have naturally, right? Because there's multiple hosts, there's multiple beetle vectors, and I wouldn't really think this as a viable solution. However, if we could understand the mechanism, maybe something synthetic can be produced that can also elicit the resistance on or tolerance on these trees. So I think that my goal is to have a proof of concept and then go back, understand the mechanism behind it, and then try to come up with an alternative that is not using lauricola. But you are right, it could explain something that we are seeing not only in Florida, but in back where the center of origin is.
Thanks. One last question, and then you can answer the rest on the comments. Katerina wants to know if you notice any reduction of growth in the vaccinated plants.
So, so like fitness of the plant, yeah. So eliciting the immune response in trees has a cost, right? We know that when we use different hormones to trigger the cascade of response, it has a cost on the trees. But we still haven't looked at it. It will be, once we stick to a couple of treatments, I think that's a good idea to measure, because at the end, we are working for the growers, right? We want for them to have a sustainable option that is also economically feasible. So if we make a tree that can produce avocados, then it wouldn't make sense.
Okay, thank you. So the next speaker is going to be Jose Luis Olivares.
Oh, I have to stop sharing.
Yeah, I'm here. Just let me share my screen. Sure. Can you see the screen?
Yes.
Okay. So my name is Jose. I'm from Mexico, and I work at the Institute of Ecology. The talk is about the synthesis of some novel insecticides for the management of ambrosia beetles. Here in this slide, we can see most of the common neonicotinoids that are used actually. More of them are non-chiral or administered racemic mixture. So as a part of our research program in finding some molecules with biological activity, but also with the characteristics that these molecules has chirality, we start the synthesis of these molecules. I'm not gonna show all the details of these reactions, but please pay attention in these two molecules, 8 and 9, which contain the main characteristics of neonicotinoids, which are the nitroguanidine moiety and also this amino acid, which is the L-proline. L-proline is cheap, commercially available, and also has a lot of interesting biological activities. Once we get these two molecules, we run the experiments using Helleborus affinis as the model of study, and we compare the mortality of compound 9 and 8 with dinotefuran. We found that after 12 hours, the mortality of dinotefuran was 40%, and with the compound 9 was 93%. We also run the docking in silico study using the research cavity with this protein, and we found some interesting results. First, the affinity of compound 9 when compared with dinotefuran, we can see that compound 9 show it minus 6.57 kilocalories per mole, and dinotefuran minus 5.64. This suggests a better affinity with the receptor, and to our delight, is in agreement with the experimental results. Also, from the picture here, we can observe an interaction cation P with the aromatic ring of compound 9, and this is well known that plays an important role in neonicotinoid insecticidal activity. Finally, the main worry when this kind of compounds are used is the toxicity. So we run the neurotoxic evaluation of compound 9, and we found that the elapsed time required for the first fall when the mice are used is higher when the rats are administered with dinotefuran. These results suggest that the compound 9 is much safer for mammalians than dinotefuran. And I think that's it. Yeah, that's it.
All right. Thank you for the presentation, Jose. Looking at the chat box right now, we don't have any questions, but please some will come. Okay, there's just one there from Daniel. It's saying, how are the beetles exposed to the insecticides?
Yeah, we run the experiment using the contact toxicity on filter paper.
All right. I also have a question. I was just thinking of the effect, say, of the insecticides on the tree itself. What are the possible effects besides the beetles? What about on the tree itself?
So you mean how this molecule can be used in the field?
Yes. No, I'm saying, can there be side effects of the insecticide on the trees that these beetles also go and attack?
Sorry, I don't understand the question.
Disculpa, qué pena. Te está preguntando que si el tamaño del insecto tiene que ver o es influenciado en la resistencia o no al insecticida. O si tiene que ver algo el tamaño del insecto.
No, I don't think the size of the insect has some impact on the results.
Okay. Then maybe one last question before we continue. Looking at the effectiveness of the insecticides, how often do you need to apply or use them?
Well, right now we don't know. Maybe it's the next step to do these experiments in the field, to know how much and how many times we need to apply this compound. But I think it's the next.
Okay. All right. Maybe I'll just allow one more. How do you envision using these insecticides in the field?
Okay, this is a good question. Well, we think that we can just prepare the solution of this compound and then put it directly into the trees.
Okay, interesting. Okay, so we'll leave it at that. And our next speaker is Jonathan Crane, who will be talking about Laurel Wilt status in avocado groves and grower-initiated control testing.
Okay, you can hear me, I assume. Okay, you can hear me?
Quietly, but yes.
Okay. Let me, is that better?
Yeah, you're good.
Good.
Okay. Okay. So I'm going to talk a little bit about the status of Laurel Wilt in the commercial avocado industry in South Florida. It is the 4th largest fruit crop in Florida. Prior to the epidemic, we had about 7,400 acres. We now estimate there's about 6,000 acres. So that's about a 19% loss in trees, actually. So the industry consists of about 430 growers, 35 handlers, a wholesale value of about 35 million, and overall impact, economic impact is about 100 million a year. So we've lost somewhere around 1,400 acres, and the estimated tree worth, tree value is about $49 million. So current recommendations, this was briefly touched on, part of it. Our recommendations include promoting tree health, because we know trees that are under stress can attract ambrosia beetles. Also to scout the groves or the orchards very frequently to detect Laurel Wilt symptomatic trees. Rogue those trees, and that means uproot and destroy the wood of the trees. To treat the immediate area around the infestation and the wood chips that are left with a registered insecticide, usually twice. And then also to suppress ambrosia beetle activity, we've been recommending treating the groves with Botanigard, which is a entomopathogenic insecticide, during late winter and early spring, because we know from the work, Daniel's work, Dr. Carrillo's work, that the beetle population, the activity of it actually ticks up, moves up during this time period. So we're trying to suppress that beetle activity. We also recently have done some work in collaboration with Dr. Carrillo's lab and looked at the effect of light levels on activity of the beetles, and found that maintaining higher light levels in a grove suppresses the beetle activity, and that's what that figure is on the bottom right-hand side. Also, we have a recommendation of prophylactically injecting trees prior to an infestation with tilt, which is propiconazole. It's a registered fungicide for injection or infusion. Has to be done about every 12 to 24 months. Mitigation practices that are currently happening. Rejuvenation, which means taking old avocado groves that are not very productive and very large trees. It's like walking into a park or a forest, and rejuvenating them. And that means cutting them back down to about 4 to 5 feet and restarting the canopy. Also, that's also used part of the technique for top working. So we know some varieties are more profitable than others. So this is an opportunity to change from less profitable to more profitable avocados, and at the same time increase the light levels in the grove. Also, we know from some surveys that somewhere between 56 and 70% of the trees being lost are now being replanted with young avocado trees. So that's in an effort to maintain production and in the industry. We also know that these younger trees are not root grafted to anything, and they are much less attractive for colonization by ambrosia beetles. We have been lucky enough to get some county, state, and federal grants for removing avocado trees and then replanting them. So to get to the plan of work for these research extension demonstrations, and Dr. Gaziz alluded to some of this, and Dr. Carrillo is also involved. It's one of the things we want to look at, and this comes from grower observation and grower practices, things we see happening in the industry. One is to look at the symptomology of and the progression of the disease based on where the pathogen is inoculated in the tree. So for instance, is it at the base of the tree, or is it high up in the tree, A1 and A2? So in A2, it's just one limb up in the upper canopy. How does the disease progress differently, and what's the reaction to it? We also want to look at the effect of implementation timing. So when you implement something called the barrier method, and that's basically removing the initial laurel wilt affected tree and then stumping the adjacent trees in an effort to stop the movement of the pathogen through the root system to adjacent trees. In addition to that, there's another method, some people call it the bagging method, which is basically a solarization method, where the trees are stumped and then covered in plastic bags for a period. It's a variable period, anywhere from a month to 3 months, in an effort to basically suppress or maybe kill the pathogen. We'll see what's going on. We've done some preliminary work on that, and we want to look at this more carefully. Another project that we've got that's going to be implemented is the effect and the timing of limb removal. So you'll see a tree like the one in A2. Can I go up and cut that limb off and stop the progression of the disease? Somewhat controversial. Sometimes it appears to work. Sometimes it doesn't appear to work. So we want to look at this in a more systematic way. We also have a small planting of avocado varieties on different rootstocks, including Haas and Lamb Haas and Lamb Haas on Duke. And we're going to just sacrifice those trees just to get a preliminary look on field-grown trees that are from the major industry, Haas and Lamb Haas, and see how they do once they're inoculated. There's been small trees where that's been done, but nothing from the field. And then also with Dr. Carrillo, comparing some entomopathogens on ambrosia beetle control. And that's it.
All right, thank you, Jonathan. And we can move on to some questions. Yuri said, when you lop off the tree canopy, doesn't the stump become much more attractive to the beetles?
Well, yeah, so originally the red bay ambrosia beetle we know is attracted to avocado and the volatiles from avocado chipping or avocado damage. But these other ambrosia beetles don't, we have not noticed, and Daniel can address this if I'm wrong, we have not noticed that that's the case, that they're attracted to the damaged wood, the injured wood.
I'll agree with that.
Okay. And then Demian said, how recommendations and mitigation tactics change for home landscapes? What are the current recommendations?
Yeah, so 2 things. We have 2 published fact sheets, and they're in English right now, one for commercial growers and one for homeowners, and it's going to be coming out in Spanish as well. Unfortunately for homeowners, they do not have access to the fungicide, and most likely they will not get access to that fungicide. The best they can do is to really keep a healthy tree, keep the trees well pruned, to keep a lot of light around the tree and in the tree to hopefully suppress its attractiveness to ambrosia beetles, which could be carrying the pathogen. That's really about it.
Thank you, Jonathan. We'll ask one last question. There was a lot of talking in the past about the interaction between the laurel wilt and the Phytophthora in grows. Are you seeing the laurel wilt as an effect, after effect of Phytophthora infection, or is it independent?
Well, and I'll leave it open for Daniel to chime in too. Basically, if you have Phytophthora root rot, and so if the water table is being held high up, you can end up stressing the tree, the root system, right? So oxygen stress. That leads to ethanol production. Ethanol production leads to attractiveness to ambrosia beetles. So that has been an issue in this area because the water table here is managed totally by man. And so sometimes it's held quite high. And we have, some feeling that that may be happening in some areas and attracting beetles and therefore an infestation. Go ahead, Daniel.
Yeah, Jerry, there's no systemic study looking at that relationship, but yeah, in groves that have a history of Phytophthora, you usually find more infection of Laurel wilt. That's is very conspicuous, a conspicuous correlation, but it hasn't been demonstrated experimentally. Or
I love how you're distinguishing correlation, observation, and evidence. I like this is amazing. Thank you, folks, for doing the amazing work down there. We have the last presenters, Xavier or Derek. I'm not sure which one of you is online, but I trust one of you is. I see Xavier. Oh. Hello.
How are you? Okay, so okay. Hey, so we have been working on developing a push-pull system for managing Laurel wilt, both in avocado and in forest. So focusing on red bay ambrosia beetles in red bay and the other beetles in avocado. So if you're not familiar, what's a push-pull? Basically, a push-pull is the combination of two first an attractant on one side and a repellent on one side. So the idea is we use the repellent to push the beetle outside the grove, outside the forest or the area where we have red bay we want to protect. And then we have an attractant that will try to pull the beetles outside the area of interest. Luckily, with the red bay ambrosia beetles and the ambrosia beetles that attacked avocado, we kind of identified a repellent and attractant. So regarding the repellent, we have been using with quite a bit of success now, verbenon, which is an anti-aggregant pheromone. And you can see here, it's kind of one of the data we had on cumulative numbers of grubbertus per trap has been published this year. It's been repeated in 3 fields, so it's pretty strong data. And here is for verbenon application. Here is our control. Methylsalicylate here was another repellent that didn't really work. But if you look at the verbenon versus the control, it's pretty clear that it repel. At least it protects the beetles for at least 3 to 4 months. And we have also an attractant. So the attractant is all the work that's been done by Paul Kendra. He has developed these alpha-copine lures, at least for the grubbertus. And for all the beetles that are in avocado, ethanol has been found to work pretty well. And we also work with the visual cues. We know that a lot of beetles will use the silhouette of the tree as an attractant, as a cues to find her host. So how did we apply verbenon? We applied the verbenon with this splat. So it's a collaboration with the company is called Iska Technology. So basically you can apply directly the repellent on the trunk like this, and that give you this kind of big dollop you can have on this. The other way you can apply the splat, you can apply by drone, you can apply by tractors. So in our experiment, we kind of apply this with this gun. We did most of our experiments in Gadsden County, in North Florida here, and Wakura here. And of course, all of the avocado has been done in collaboration with Daniel Carrillo in South Florida. So with the data, this is what we have from last year. That was the data we have on red bay, on ambrosia beetle. So we did the experiment in Wakura. So we had a control here, red bay trees that are not treated with anything. And we are looking at the capture of Grabatus on a white sticky trap. And you can see that on untreated control, the capture of Grabatus is pretty high around the season. With the push-pull treatment here, you have the pull only, so the attractant only put on the outside of the area we wanted to protect was the less efficient. That's what we can expect. And the push and the pull were very efficient until we have this Hurricane Michael hit the area. And we noticed, and you probably noticed this too, after an hurricane, we have all this Ambrosia beetles population that increase. And at this time, we were not able to protect the trees anymore. We only treated the tree at the beginning of the experiment, so it's also 3 months of protection just with a single application. It's kind of interesting. And in this case, so what we only used the alpha-copine as the attractant. And last year, in Lake Tarquin, which is just nearby Tallahassee, we used the tree silhouette. So it's a fake tree basically that we use, and we add an attractant on it. And if you do that, we have the control here. We have less Grabatus in this case, but nevertheless, we had the peaks here of Grabatus. And the interesting thing is the push-pull, we didn't have too much capture of beetles on our push-pull treatment. We had a little peak on the push only, but not on the push-pull, as you can see here. So pretty long protection after 11 weeks. And if you're wondering how the tree silhouette is attractive to beetles, this kind of a small experiment we did. We have the red bay here, and we compare this to our factory. So the factory is really, it's a pole, a cardboard, cylindrical cardboard, and we put an attractive color on it with lures of alpha-copine in this case. And you can see the capture of beetles on our factory is much higher than what we will capture on a red bay tree. So really drive the beetles on this factory. So that works pretty well. And that's the data we have on Ambrosia beetles. This has been published this year in scientific reports. We kind of did the same system and a push-pull with red bay, but in this case, we only use ethanol as the attractant. We were on the process to use the factory too this year on avocado, but then we had to stay home. So we will wait a little bit on that. But anyway, what you can see here, we had 3 weeks of experiment. We have trapped very close to the avocado, so less than 1 meter, and trapped that were put a little bit further than the like 1 meter away from the avocado tree. If you look at the trap away from the avocado tree, you don't see too much effect on the treatment. However, if you look at the sticky trap we put on the avocado tree, you see that the push-pull system were very efficient. We didn't catch any beetles in the avocado grove on the push-pull treatment, which was quite interesting. Of course, you know that we don't have Grabatus in the, oops, sorry. We don't have Grabatus in the, I don't know what I did, but anyway. We don't have Grabatus in the avocado grove, but we have Sexeni Fornicatus affinis. That was the main species we found. So with this, I'd like to thank all my collaborators, a lot of them from UF mostly, but also Monique Rivera from UC Riverside. Do you have any question?
Thank you, Xavier. Great. It's awesome. One question. I've heard that the splat technology tends to melt in Florida summers. Is that true, or are you using a different formulation?
No. I mean, you know, you need to reapply your splats. I mean, if it's very hot, obviously, and very humid, you will have less efficacy. So I will not say it melt completely, but obviously, the warmer it is, the shorter your duration period is. So instead, we did most of our experiments, if I recall, in fall. So when the temperature is starting to drop, so we had 3 to 4 months efficacy. I'm not sure what would be the efficacy during a very hot summer. probably something to look at, but it would be probably shorter. You probably will need to apply more.
I can kind of comment on that.
Yeah, good.
Basically, during the fall, winter, and early spring, the splat dollops would last upwards of plus 4 months. In the summertime, I found myself having to replace the dollops after about 2 months. It seemed that the amount of humidity and average rainfall seemed to affect it the most. Basically in the summertime, when we start getting those daily showers, that's what seems to affect the decay of the splat the most. But again, that's during a 3-month period of the year versus the remainder of the year where the weather is slightly more stable. I noticed that the splat did pretty well, particularly because the red bay trees are a little bit more of a canopy tree. So the splats kind of get protected by the trees themselves. However, during the summertime when we had very hot and humid weather, they did tend to break down rather quickly.
Thank you, folks. This concludes our meeting. I think everybody, this was amazing. Maybe one day we'll do that again. Before we all conclude and leave completely, I would love to ask you to turn on your camera and smile, because I will take a screenshot so that we can post it everywhere. Yeah. So, hello everyone. And oh, there's the youngest member of the team is with us. Thanks a bunch, everyone, and have a great day. Done. Bye.
Bye.
Thanks.